Good morning, everyone, and thanks for attending this seminar. For the person that are in presence and for the online attendant, thank you very much. And I am pleased to have organized this seminar. This seminar was funded by the Universitas Mercatorum project, TSP-ARC, topological signal processing and application to brain and knowledge networks. And the goal, the aim of this seminar is not only to present the result of the project, but also to explore in an interdisciplinary perspective different approach to the study of brain network and understanding how brain organize its connection and how these connection interact among them is a very fascinating and interesting research topic. And it requires, of course, innovative tools for the study. And an important and key point is that it also requires an interdisciplinary collaboration among different researchers with different skills, because understanding and study the brain require skills from neuroscience, from engineering, from machine learning expert, biologists, and so on. Well, I think that we can start. I, of course, thanks all the speakers for their contribution to this seminar. I will start with my presentation. In this talk, I will introduce topological signal processing and some advances for brain network analysis. Well, the goal is to extract information from data. Data often reside of irregular domains that are not Euclidean, but this domain have a structure, so that the goal is to extract information from data, and in particular learn, discover the interconnection among the elements of the network that we are analyzing. We can go to the next. Okay, one of the main goal, one of the main challenge is represent data of a domain that is able to reflect the connectivity of the data. What kind of space we can use? We can use topological space. Topological space are the more general type of space. We have here some example of space: manifold, graph, simplicial, self-complex. A topological space is simply a set of elements with an additional structure that is called topology. Topology is a set of rules that is able to express the connectivity among the elements of the set. Can you go next? And graph-based representation are the most usually used representation to analyze data. In graph, data or signal are associated with the nodes of a graph, and we have a link between two nodes if there is some kind of relation between these two data. For example, in brain graph, we have different ROI region, and we can have a link between this region if there is some sort of similarity between the data that we are observing. In DNA nucleosome graph, we have a protein, and the protein associated with the nodes, and there is a link between two proteins if, for example, there is a reaction among them. We can go to the next. And well, the most important step is provide topological space with algebraic operators that are able to reflect the connectivity of the space. This operator, the most famous operator, is the Laplacian matrix. The Laplacian matrix is defined as the product between B1 transpose, where B1 is a matrix reflecting the node edge incidence among these elements. In particular, if we see this graph, for example, the first column A1 describe the endpoint of edge A1. These endpoints are V1 and V3, and so that we have 1 minus 1 in correspondence of these vertices and 0 otherwise. Then we have this simple representation of the graph. We can go next. Okay, in graph signal processing, we enable the processing of a signal defined over the nodes of the graph. Well, what is a signal defined on the nodes of a graph? It's a simple mapping from the set of nodes to the set of real or complex vector of dimension n. N is the number of nodes in the graph. Then we have a vector, and each entry of this vector is associated with the nodes and represent the signal over this node. Graph signal processing extend all the tools that we know for the processing of a signal to the processing of a signal over graph. Then we have a spectral representation, filtering, sampling of data, and recovering from this sample the signal over all the network, topology inference, learning the structure of the graph from data, and so on. We can go to the next. To represent, to enable processing, we need to represent the signal over a given space. We need a basis to represent the signal. Then what are the more suitable bases to represent the signal are the eigenvector of the Laplacian, because these eigenvectors are able to reflect the connectivity of the domain. Then we can project with a simple linear product. We have a linear algebraic operator in this case, and we can simply project the signal over the eigenvector of the Laplacian, and we get a set of coefficients. We can represent this signal over, okay? We can represent the signal by using, for example, in this simple graph, by using 3 coefficients. Okay, then we have a sparse representation of the signal. We can go. Okay, now graph signal processing is useful for representing node signal on the nodes, and is able to capture pairwise relation. But in many systems, we have a complex interaction among data, and this interaction often involve group of entities, group of elements, not only 2 nodes with a link, but we have a group of nodes that are involved in a given relationship. What is the challenge? The challenge is capturing a multi-way relation among data, and of course also use topological domains that are able to represent this higher order interaction among the data. We need to go beyond the graph. We need higher order topological space, such as, for example, simplicial or self-complex. If we see this figure, we have the simplicial complex. The simplicial complex is different from a graph because we can see also the presence of triangles if we see this figure. This means that we use a triangle to represent a relation between three nodes. Then, if there is a relation between the three nodes, we fill the triangle. If the triangle is empty, there is no relation between these three nodes. Okay. In cell complex, instead, we have also square polygonal cell. This means that we are able to capture different relation among the nodes. Can we go to the next slide? Okay, topological signal processing is a framework that I developed with Professor Sergio Barbarossa in this published journal, and it simply applies signal processing to signal defined over topological domain. We can represent the signal of this domain, or we can analyze the signal, we can enable the processing of signal, filtering, sampling, recovering, and so on. You can go to the next. Okay, an example to better understand this higher order interaction. For example, we have a co-authorship network. Vertices are authors. There is a link if two authors have at least one joint paper. And there is a triangle between the three authors if they have co-authored at least one joint paper. If I use a graph, I am not able to express this relation among three authors because the graph is able to capture a pairwise relation. Can we go to the next? Okay, and this is the definition of simplicial complex. It is a finite collection of a subset. These subsets are called simplicious of various cardinality of the element of a set V. And an important property of this simplicial complex is that if I have a set of nodes, all the subset of this set also belong to the complex. For example, in this graph, I have nodes. I name node 0 simplicious of order 0. I have a edge. I name this edge 1 simplicious of order 1, and these are able to capture pairwise relation. And I name filled triangles 2 simplicious of order 2. These are able to capture triple-wise relation among three nodes. But we have only triangles. So we can also have higher order structure, tetrahedral, and so on. But using always triangles. Good. What are the limits of a simplicial complex? In many applications, the inclusion constraint on the subset is too stringent. For example, if I have a DNA nucleosome, a reaction among a group of proteins does not imply that a reaction also occurs among any subset of this protein. Okay, then we need use different simplicial that we name cell, and these are the so-called cell complex, where we have the possibility to use polygons of different order. In this case, we name vertices 0 cell, edge 1 cell, all polygons 2 cell. It is very important because, for example, if we consider this triangle V1, V2, or V4, and V9, in this triangle, there is no connection between the node V1 and the V4, okay? And so that we are able, by using a square and polygons of different order, to capture more sparse relation among the elements of the set. If I have a simplicial complex, I have no square, I have no polygon, I am not able to fill the relation among these 4 nodes here. Okay, next. But another step is provide an algebraic operator to represent simplicial set complex to enable the processing to represent the signal. And we define a set of incidence matrix BK that are able to capture the incidence between cells of different order. For example, we have these different Laplacian of different order. Let us start from L0. L0 is the Laplacian associated with the graph, okay? If, I can represent a signal defined over the node of the graph. If I go beyond, I have, for example, cell or simplicial complex. In this case, I define a Laplacian L1 that is defined in the space of the edge of the complex. And it is able to capture the relation among edge and nodes as for graph. But it is able to capture also the relation between edge and triangles and cell and so on. The second term, B2 transpose, takes in account the incidence between edge and polygons. Okay, we need to specify this incidence. If I need to construct the simplicial complex, I need to specify the incidence between edge and polygons. The boundary of each polygon has to be specified, so that I have this second term, B2 transpose. I can go to higher order structure. We have a different Laplacian, and we name the first part down Laplacian, the Laplacian that taken account of the incidence between the edge, for example, and the node. And the upper Laplacian, the incidence between the edge and the upper cell that are the polygons. It is very interesting to remark that these two metrics, B1 and B2, belongs to orthogonal subspace. It is very important for the processing of a signal. Well, we can represent over this complex signal. We can define signal over the nodes, flow signal over the edge, and the signal associated with the cell. For example, in the example of a co-authorship network, I can associate to the cell the number of paper that 4 authors have write jointly. Okay. I think that it is a different perspective, because with a graph we can only analyze graph signal on the nodes. With simplicial complex, we have signal on the nodes, signal on the edge, flow, and the signal on the cell. We can represent signal by projecting this signal over the eigenvector of the Laplacian. It is important also to remark that there is an analogy between the Hodge Laplacian and the physics flow and electromagnetic field, because the two parts that I name low Laplacian and upper Laplacian live in two orthogonal subspace, and these subspace are called the rotational and the solenoidal space, in analogy with the physics electromagnetic field, where we have an irrotational and solenoidal part. What means an irrotational flow? Means that I am able to identify in the network nodes that are source or sink of information. Because the flow is irrotational in a node, if it is not conservative on the nodes, it means that we have that the inflow is different from the outflow. And this means that the nodes act act as a source or a sink for the flow. This is the rotational part. For the solenital part, instead, it means that we are taking account of the circulation of the flow around the polygonal cell, the second order cell. Okay, how the flow is circulating around. And then there is another part of the signal that I name harmonic component. It is very important because it lives in the kernel of the Laplacian, and the kernel of the Laplacian give us some important property regarding the topology of the space, because the dimension of the kernel is equal to the number of cavity, to the number of holes that we have in the complex. So that each of these components gives us a different feature of the space. We can go to the next. Okay, we generalize graph signal process to simplicial and cell complex. I don't go in details, but there are the journal. Some applications can be brain, transportation, water distribution network, filtering, social network, and so on. The next, we also apply to neural architecture this framework and in this paper. Okay, we can go to the next slide. Well, how can you use this frame in brain network? The challenge is in brain network is provide insight in the complex interaction among the raw region, and this is important for the study of the signal, but also for the study of diseaser, brain diseaser. In brain, we have usually functional and structural graph, and we have this kind of connectivity that can be captured by using graph. Usually, graph are used for the study of brain network. The brain connectivity is very important because the cognitive capacity are strongly related to how brain regions are connected. It is a very important aspect because in many studies they discover that, for example, in mammifers, the neurons have a similar structure in all the mammifers. But the difference between the brain of a mouse and the brain of a human is the connectivity of the network. It is very important because the neurons are similar. The size is not so important for cognitive activity, but the connectivity is very important for cognitive capacity. Next slide. Only to give you a simple example of the fact that the connectivity is able to reflect also the disorder in the brain. We have this application where we consider some study, an epilepsy study. We have some signal from these individuals that are affected by epilepsy, and we have this electrocortical signal. These signals are observed over some electrodes, and the goal is to recover the graph from the observation of this electrocortical signal by observing the individual in two different phases. In the phase before the onset of a seizure, and in the phase after the seizure. Okay, and we here recovered the graph, and by using a method that you can find in this paper. And it was very interesting to see that before the onset of a seizure, the graph is very connected. It is a complete graph. It is strongly connected. And after the onset of a seizure, there is a loss of connectivity in the graph. The graph is more sparse. Can you touch the second figure? Because there is a movie. Okay. You can see that before the onset, it is very connected. After the onset of a seizure, the graph become less connected. This means the connectivity is able to reflect this disorder. Maybe there is a loss of correlation among the signal, and anyway, the graph representation is able to capture this behavior. Next. Okay, but we have graph. We introduce topological signal processing, and then in brain network, it is very important to capture higher order in relation among brain region or neurons, because these regions are working together to process information. Working together to process information means that we need to apply topological signal processing. Need to process information over this higher order topological space. We have a signal over the node, signal over the edge. We have these triangles and the square that are able to capture relation among a group of 3, 4, and so on nodes. Can we move to the next? We propose some method to learn the brain graph, the brain cell complex, to find the sparse representation of the signal, to apply a sampling theory to recover the signal, and also strategy to jointly learn the simplicial complex and the sparse spectral representation. This result will be discussed by Dr. Breno Bispo in the next talk. And here, a simple example. I consider this data set for resting state functional MRI from the UCLA connectivity database, and I use this signal to recover first the graph and then the cell complex structure. We have this triple-wise relation and relation among the square in this figure, and these second-order cells, these polygons, are able to represent the circulation of information within this region. Here, we can also skip this slide only to give you, to say that the cell complex that we infer is meaningful. Then we recover the signal to check if there is a small mean square error between the true signal and the recovered signal. We can go. Well, when I study brain network, I understand that anyway there are some problems with this kind of network, because they are complex network. Means that maybe we need some advance in topological signal processing. We have this paper, the first has been submitted to transaction signal processing, and the second paper has been presented in September at the USIPCO conference. And what is the problem? The problem is that we have a complex network. Then maybe in complex network we have different subnets that are interconnected among them. This means that for each subnet, maybe we have different signal, and also the interaction among different subnets can involve signal that lives in different domain. The flow can be different between these networks. Then in brain network, for example, the interaction can occur at a different scale, so that we need an advance in this framework. by taking account of the multi-layer structure of this network, and by also taking account that we need to learn higher order interaction among the data. Remark is that a key challenge brain network is learning intermodal connectivity and multi-scale interaction. We introduced this new space, cell multi-complex space, that are able to capture higher order interaction among different networks. We use an operator that is named cross-Laplacian to represent this space. And we extend the topological signal processing to this space by enabling the local processing of a signal. The next slide. This is the space that we introduce that are named cell multi-complex. We have different subnet. Each subnet can be represented by using a cell complex, but also the interaction among these subnet, this layer, are represented by using higher order topological space. In this case, we have, for example, the presence of nodes of edge, but also the presence of polygonal cell between different layers to capture this higher order interaction. We can go to the next slide. What is important in this framework is that we can analyze a complex network by using a cell complex and using a global perspective of the space. Okay, if we use a global perspective of the space, we can simply apply topological signal processing to this global structure. But when we need to extract local invariant of the space, or for example, we need to investigate how layer, how subnet affect and controls another subnet, we need use this cell multi-complex space. We have a lens to analyze the simplicial complex by using different resolution levels. We can have for each layer, we can look at each layer from its point of view through the lens of other layers or as a part of all aggregate structure. We can imagine that we have two different perspectives. If I am an observer of a given layer, I am observing the other layer, but my view is different from the view of the layer, from the other layer towards me. It is different. We have different perspective according to the observer. If the observer, we can go to the next slide. In this slide, for example, I have these two layers, layer L and layer M. If I am an observer on the first layer, on layer N, I have a view of the topology that is different from an observing living on the second layer. This is a way to have a lens on the topology and the signal representation can go. Of course, these Laplacian, these cross Laplacian that are very important to represent this signal can give as a way to represent the signal of this space and to capture different property of the signal. For example, I am considered these two layers. I can identify some nodes that are called hub node, or nodes that have a control function towards the nodes of the other layer. Because these operators are able to identify cones. Cones are empty triangles between two layers, and these cones are useful to identify this cross up. I don't go in details, but only to give you an idea of this of this framework. So we can go to the next slide. Can go to the next. Here we can extend the topological signal processing to these new cross Laplacian operators, so that we can represent local signal by taking account of the topology and by using different lengths of resolution. Also in this case, we have the decomposition, the signal in different components that are orthogonal. Each component is able to capture the divergence of, in a node, or the curl, the circulation in a node, so that we have a very nice property that we are able to learn from the signal and from the space. The next. This is my last slide, but I leave the presentation to Breno, only to give you a result on the application of this topological signal processing over cross multi-complex cell. And here we considered the brain network, and we consider two layers. In the first layer, we have two subnet, the limbic and the frontoparietal subnet. And the second layer, instead, we have the default mode networks. By applying this framework, we identify some nodes that we named cross-hub that are critical for the flow between these two networks. And removing these nodes is critical for the connectivity among these two layers. But the next slide. Only to conclude, I think that topological signal processing and the recent tools that we developed for cell multi-complex space are very important for the analysis of complex network in general, but for the analysis of brain network in particular. We can, for example, in future discover multi-scale interaction of brain network, and we can extract information at the different resolution scales. Okay, thanks. I think that maybe we can move to the next. Introduce the next speaker is Dr. Breno Cordero Bispo. He is a PhD candidate in electrical engineering at the Federal University of Pernambuco, Brazil, supervised by Professor Giuliano Lima. He received his Bachelor Science and Master Science degree from the same university, where he worked on biomedical sensing, embedded system, and Internet of Things application. Currently is visiting researchers at the University of Amsterdam, co-supervised by Professor Fernando Santos. His current research focuses on topological and upper graph signal processing, computational neuroscience, and complex system. He is currently involved in the FINRIC TSP ARC project of University Mercatorum. He is working with me on this project.
Just testing this is on. Is pointer. Okay. So good morning, everyone. Here in my presentation, I will talk a little bit of the application of the TSP framework into the brain networks. So okay, let me. Okay. In the computational neuroscience field, most of the signals the studies is using is called MRI, because most of them, because the MRI has a very good spatial resolution, so you can better represent the brain network using these kind of data. So, but in the MRI signals, the two major modalities that the studies are using is the fusion MRI that encodes the structural connectome of the brain, and or they use the functional MRI that encodes the functional connectome of the brain. So, and basically using these two complementary signals, you can apply signal processing over brain networks. So, as Stefania already introduced, the GSP framework, basically this framework was invented 10 years ago. That is a natural framework to process signal over brain networks. Okay, so using this GSP framework, basically we can characterize any irregular domain such as graph. So in the neuroscience, we can represent a brain network using a adjacent matrix that can be constructed based on the diffusion MRI or the functional MRI using statistical metrics or any other metric that you can build the brain networks. So we have the graph, that's the domain, and also we have the brain signals. What are the signals of the node of on the nodes of the brain networks can be the fMRI, for example, the fMRI signals. There are this time series, these signals over each node of the brain. So we have algebraic operator that characterize the pairwise similarities of the brain network, that's the Laplacian operator, and applying the eigendecomposition, you can synthesize or you can represent any brain signals based on these eigenmodes or brain harmonics. That these harmonics can encode any functional connectivities in the brain based on this figure. So you can also apply the graph Fourier transform to analyze the spectral domain of these brain signals. So the GSP is already consolidated in the neuroscience field. For example, here on the first figure on the left, you can the work from Van der Wiele can demonstrate that any graph signal can be decomposed into these graph harmonics. You can characterize coupling and decoupling connectivities on the brain, and also on the right figure there is our application into that's a work from Farrugia that helps the GSP framework helps to characterize or to classify healthy subjects to subjects with Alzheimer or schizophrenia, etc. That's a few applications of the GSP. So basically, the graph are able to capture only pairwise similarities between signals over nodes, but as Professor Stefania mentioned, the simplicial complex generalize this into a higher to capture a higher order simultaneous interdependency between signals over simplices. So we have naturally the brain doesn't work only pairwise connectivities. The neurons, for example, they can communicate to multiples neurons, etc. This the simplicial complex or cell complex are natural representation of these high order connectivities in the brain. So the topological signal can be are defined over the simplices. So the S0 are the signals over the nodes, the S1 are signals over the edges, the S2 are signals over the triangles, etc. You can extend this to a higher order algebraic object. So the algebraic representation of the simplicial complex are use the Hodge Laplacian. You can represent the brain Hodge Laplacian using this framework, as Professor Stefania mentioned. So after we define the suitable algebraic representation of the simplicial complex, you can also decompose the Laplacian into the three orthogonal subspaces: the gradient, the harmonic, and the curve subspaces. That any simplicial signal can be decomposed into three independent flows. In this slide, in our case of study, we only focus on the edge domain simplicial complex. And also the topological operator that the TSP framework provides us, the diversity in the core operator, help us to better understand this functional connectivities of the brain. So,
It works.
The movie is not working. I don't know why, but yeah. Okay. So here is just a few examples just to demonstrate there's a preliminary result that we obtained. So how this orthogonal or the gradient subspace of the brain topology can give us which kind of information. So the low frequency flows of our brain topology can give us information about the connectivity between the subcortical areas and the visual areas. The middle frequency flows can encode connectivities between the visual cortex and the DMN, the default mode network, and the high frequency flows can encode information that are exchanged between the visual cortex and the motor cortex, for example. And also, if we apply the divergence, you can identify which nodes are spreading information, that's in the blue dots, or are sourcing information, that are aggregating information. So this kind of topological operator is very useful to identify the source and sink of information. And also applying the curl, analyzing the curl subspaces of our brain topology, you can also identify flows that are circulating among the different nodes that are in different nodes that belongs to different brain subnetworks. For example, on low frequency flows, we have here information that are circulating across the visual cortex on the left, the first thing on the left, the visual cortex and the motor cortex. On the middle frequency, we have flows that are circulating mainly among the ROIs of the visual cortex and all the ROIs of the other brain subnetworks. And for the high frequency flows, we have here flows that are circulating between the DMN, or the default mode network, and the frontoparietal network. This example is just to a preliminary result to show the potential of the TSP framework to identify or to characterize brain functional connectivity. So we have the raw fMRI time series. Our nodes for our framework, there are nodes over the are signal over the nodes, but we can also define what are the nodes over the edges to apply this TSP framework and analyze the functional connectivity on the edge domain. So we can use the co-fluctuation method that basically is the element-wise multiplication between the time series. So we can define the edge signal based on that's represent the first order of fluctuation, and then we can apply our approaches. So first, what in this work is a couple of results that we obtained in the paper published in the OCIPCO in this year. So basically, we apply these methods into the resting state fMRI data from the Human Connectome project. using 300 unrelated healthy subjects using the Schaefer Atlas. So we first infer the brain topology, so the brain skeleton of our brain topology using Pearson correlation. And then after we built the brain skeleton, we infer the field triangles or the high order interaction of our brain topology. The first approach, we used statistical multivariate information metrics called total correlation to select the most important field triangles. And also our second approach, we can use optimization problem that's based on the minimization of the circulation engine along all triangles and the data fitting error. So I will not spend a lot of time here, but basically it's an optimization problem to infer the high order interaction of the brain after we built given the brain skeleton or the brain graph. So we apply the basic topological operators such as diversion over signals from the HCP data set. So we have here our first result that we identify that on the left hemisphere, they basically are the major sink of information, especially the visual and DMN and the dorsal attention network are spreading information to the right hemisphere of the brain. That's specifically to the right hemisphere that belongs to the visual cortex, the ventral attention, and the DMN subnetwork. And then we apply the curl operator over these same edge signals on the brain, and we identify interesting results, especially when we analyze the conservative circulation patterns, that these triangles, this circulation, they remind us the idea of segregation, integration of the brain. So we have here triangles that are circulating inside or along only with the visual cortex, and we have circulation that are among only with the motor cortex. That this reminds us the idea of segregation, functional segregation, and there are triangles that are circulating among among the visual ROIs and the motor cortex. So after this, like we applied the recent framework developed by Stefania and I and Fernando, that's the TSP cell multi-complex framework. I will not spend a lot of time here because Stefania already introduced this framework. Basically, we already defined what are the cell complex, what's the main idea, what are the algebraic operator used to analyze the cell complex between layers of a given example. In our example will be the brain networks. So we have the hot decomposition, we have the topological operator, all the same framework used here is already introduced in the last previous speech. So first we need, how can we apply this framework to the brain network? First, we define which ROIs or which region we would like to analyze. We select 3 subnetworks. The first frontoparietal and the limbic network represent we assign these to subnetwork is our first layer of our PSP CMC framework. So our first layer composed by 18 nodes or 18 nodes, 80 ROIs that belongs to the limbic and the frontoparietal subnetwork, and the second layer is our DMN, is our ROIs that belongs to the DMN subnetwork. And then after we define which is the first layer and the second layer, we built we build the cross edges. These cross edges were constructed based on the precision matrix of our usually. So given the edge signals, given the node signals, we use the precision matrix to infer the cross edges, and then we have these results that on the first layer we have the FP and the link subnetwork, and the second layer the DMN, and these edges between these two layers are our cross edges. So after we define the brain multi-complex skeleton, so we need to define the high order interaction that's given this brain skeleton, and these cross cells are inferred using the optimization problem. That's the goal is to minimize the variation of the cross edge signals. That case is the smoothness of the signals onto the upper cross Laplace angle vector. After we solve this optimization problem, we have a complete setup of our cross multiplex using brain data set. So it's given by this. So we have in total 34 field cross cells. So 29 blue triangles and 5 orange quadrilaterals. So after we define our brain setup, we apply the concept of brain cross network hubs. Basically, we identify which nodes are crucial or very important that controls the other layers. For example, we quantify the strength of the harmonic cross hubs. So we analyze which node of the second layer, so the DM that belongs to the DMN subnetwork, controls the first layer that's it's composed by the DFP and the limbic subsystem. So we, in these black dots, as you can see, and the figure, represent the cross hub harmonic intensity. So the most intense cross hubs, and these here you can see the main cross hub that controls the first layer. So that's one of my main results. So, and after this, so we conclude that specific DMN nodes modulate and coordinate activity across networks in the first layer, as I said. And this result aligns with established results in the neuroscience lectures, because this is already known that DMN, some ROIs from the DMN are act as a connector hub, enabling direct, indirect connectivity between the frontoparietal network and the link network during the resting state. So after we proceed with analyzing the cross circulation patterns and the network integration, so we apply simple call operator over these cross edge signals. And after we apply this, we analyze the most significant cross circulation, and we reported that strong circulation, that's representing strong green quadruples, they show us a strong functional coupling between the DMN and the frontoparietal network. This reflects the integration between the DMN and the FPN during resting state. So, and these results support the idea of internally oriented cognitive process, so such as autobiographical memory, planning, self-referential thinking, etc. And also, if you analyze the most, like the pronounced negative cross circulation, that's representing strong pink quadrilaterals or triangles, we notice that this if pronounced negative cross circulation are circulating among the ROIs from the limbic and the FPN and DMN subnetwork. So this indicates functional integration across the emotional and cognitive control systems, such as the limbic and the FPN, and mediated by the DMN network. So, and this result is consistent with prior studies highlighting that DMN is act as a major role connector between the limbic and the FPN subnetwork. So, summarizing, so this provides interpretable so we proposed methods to ensure the multi-layer brain networks, and after we apply the crucial, after we apply the DSP CMC operators, we identify hubs and crucial cross circulation among ROIs of the brain networks. So that's all.
Okay, thanks, Breno. Okay, I introduce the next speaker is Professor Fernando Nobrega Sancho. He's an assistant professor at the Northwest Briggs Institute for Mathematics and the Dutch Institute for Emergent Phenomena at the University of Amsterdam, where he is also affiliated with the Institute for Advanced Study. His research bridges theoretical physics, applied mathematics, and data science, with a focus on applying tools from topology, geometry, and statistical mechanics to network, neuroscience, and complex systems. He has previously held the research position at Oxford University, the International Center for Theoretical Physics, the Amsterdam University Medical Center, the Federal University of Pernambuco in Brazil. Beyond his research, he is committed to teaching and mentoring, aiming to inspire young scientists by highlighting the interdisciplinary power of abstract mathematical concepts in understanding complex real-world phenomena. He is also currently involved in the FIRIC TSP ARC project, and he will hold the talk Interdependent Scaling Exponent in the Human Brain.
Thanks. Thanks, Stefania, for a nice introduction.
Okay.
Also, thanks for inviting us and for organizing this nice event. I'm very happy to be here. Yeah, so I prepared a topic that it's related to topological signal process, but in my view, because the title of this event, a cutting edge in perspective, we have a good perspective on the signal processing on coarse graining of networks, and that's what I'm trying to discuss for today. Okay, wait for the slides. Yep. The title of the talk is Interdependent Scaling Exponents in the Human Brain. It relates to our old topic in complex systems, or also in fractality, etc. So you have a definition of scaling variance. So I will. Okay, this is working. Or? Yeah. So you can go. Yeah. So I will talk about what scaling variance and what hair normalization means, but in a naive perspective, when you coarse grain variables, and why I'm talking to that. So Stefan and Breno already explained, data is everywhere. Data is available. We need to get information from the data. but sometimes data is not the right resolution. Data is coarse-grained, and the brain is one typical example of that. Like we know that in a human brain we have approximately 80 billion neurons, but when you observe the brain, we don't observe the brain itself. We observe an image of the brain at specific time resolution and spatial resolution. So then in MRI that Breno mentioned, for instance, the resolution you have spatially is only 1 cubic millimeter, that you have approximately 100,000 neurons, order of magnitude. So you already lose a lot in the data. So it's not, when you have the best data, the perfect data, then you have the best theory, which I think TSP is among them. But sometimes your data is already coarse-grained from start. And then that also happens when you cross scales. So we have like from atoms to cells, to molecules to cells, to tissues, to the full body, to behavior. So all these scales need to be integrated. So in a way, one perspective I want to bring for today is that how can signals propagate across scales when you have a limited resolution on the scales, or when you coarse-grain the scales itself as a procedure. And that's why in physics, so in the, I'd say 60s to 80s, it was a hot topic in physics, a renormalization group, which means mechanistic, like a way of coarse-grain networks, or not networks, physical systems. And then very recently, this is being translated to networks. So I will then talk about how we did that for MRI and which surprise we got when we did that for MRI. And that brings my perspective that we may need to do a TSP kind of theory for coarse-graining as well. So you can get, yeah. So in a historic perspective, what I want to bring is that when you get this type of coarse-graining, you have a property that's called scaling invariance. That's is common to know when you see a fractal. Like in a fractal, every time you zoom in a fractal, you still see the same thing. So then if you have like a Mandelbrot set or any type of fractal, every time you zoom in a fractal, you see the same property. And this property is called broadly, naively scaling. And what is interesting is that in the early last century, there was this scaling across variables around a critical point, that when you have a phase transition in a physical system and you observe like a specific heat curves or susceptibility curves on phase transitions, and you would make a log-log plot of those measurements, you would find the scaling invariance. And on that, you could measure for different systems this scaling invariance, and there was a property called universality. So no matter the material, if the material belongs to the same universality, they would have the same scaling properties. And that was a huge development from beginning of last century until nowadays. People still use it, but let's say the peak was when there was a Nobel Prize given by one of the inventors of the theory in renormalization group in the 80s. I think it was 84 or so, by Wilson. And why I'm talking about this history is that back then, this was made in a theoretical level. So you would get a model of particle interacting, and you would think, oh, how I coarse-grain those particles like spins, how I can coarse-grain spins, like merge spin variables, and how this is preserved or not when you coarse-grain the variables. And nowadays, because you have enough data, you can also do that with real data sets. We can think about the brain, oh, I can observe the brain across all the voxels, but I could also coarse-grain the voxel, average the neighbor voxels, and get a more coarse resolution of the brain, and do it again and do it again until I have only a small number of areas in the brain. I could also do the other way around, but for that we don't have enough resolution yet. Because if you would think of the smallest unit in the brain, which is not necessarily true, but is consolidated as possible, would be a neuron. You would have 80 billion units in the human brain, and you would need to then make a procedure where you could coarse-grain one neuron at a time. You get 80 billion neurons, but you could check how pairs of neurons fire. Then you would have half of them, 40 billion, and then how quadruplets of neurons fire, and you would have a half of half. So you could go not so far, because this goes exponentially, but if you go 35 times or so, you would get the 80 billion neurons. And that's a procedure that empirically would be a cost graining your variables. And that was developed like in the 80s, and now people, lots of Italian scholars are developing this, also in Netherlands, in Lucca, on how to do this procedure formally within a network. Yeah, so that's kind of a historical perspective. You can go, and then I will go to some definitions and then what we did in the brain. So basically, scaling variance, as I already said, and I put the idea of the fractal, is that when you're rescaling a system by a certain factor, the system looks the same. And that is a signature of that is a power law factor. Every time you have a observable in a certain interval where a property goes with a power law, you have this property. When you rescale, you have a characteristic, no characteristic scale, so the ratios between your system when you zoom in or zoom out becomes the same, as you see in this fractal tree. And then one way to observe this experimentally is the log-log rule. So if you would have a power law, through different measurements you have in a log-log scale would behave like a straight line. Of course, it's more how to find this. It's a naive. You would need to see it to several length scales to really make sure this is true. Because if you have limited data, you could have a pseudo or not real scaling variance, but that's kind of in a naive way. You can go to next. That's how we would define. And these have appear near phase transitions, as I told in the beginning, in fractal processes, growth processes, and in complex systems. There are some caveats. It's not easy to measure. If real data are finite, in fractals, theoretically, you can do it as many times as you want. But that's a limitation what you have. We live in finite world. If you believe in quantum mechanics, everything has a finite moment. But at some point, things become finite. So, but basically, the idea is that now with the availability of data, you could do this procedure to complex data sets. And that this project, we did this procedure with brain networks with MRI data sets. And okay, I'm anticipating what's going on. I'll go a little bit more in the theory. So one more. Oh, sorry. Yeah, then basically that relates to one topic of brain network analysis called criticality, which is, we don't use it here, but we can assume that the brain, because of this scaling variance, and scaling variance was observed around critical points of phase transitions in the past, as I just mentioned, that the brain may operate in a dynamical critical point, around the bifurcation point. And that also has an interesting property, that the correlations across all scales are very strong. If you are in a bifurcation point, in a critical point around the phase transition, the correlation across length scales are strong. This is only a hypothesis. I didn't use it here, but it's good to give a context where this is framed. And of course, this may not happen, but may happen. That there's a community in brain scholarship that work under this umbrella. You can continue. Yeah, so then I put this idea that was summarized by this book of Dom. So if you see the top figure in the right, you see like a specific hit. So is a plot from the, I don't know, 20s or 30s, that you have a material, and then you hit the material, and a phase transition, you melt the specific heat, and then after phase transition, the specific heat decreases. And when you do a log-log plot, the neighborhood of this phase transition, you see that's a signature of scaling, that you have a linear relation between the log plot of the two variables. That is the beginning, and from that we can continue from where we came. And what that means operationally. Now let's go more to the maths. That means that if you would have, like in this specific case, I go to a spin system. If I would have a spin system, I would have a spin variables, and then I can create a rule to cost grain the variables. I will just use the simplest rules that the majority voter rule, or the neighbors rule. Oh, let's say I'm up, so if all my neighbors are up, I continue up. Or I select a subsample of the space, and I when I cross-grain, instead of representing the space in a high resolution, I just exchange all those variables by the one that's most present around them. Let's say in this top circle, you count how many arrows are up, how many are down, and you substitute all those arrows by one unique arrow that is just the majority. So that's the way you would cross-grain. And what happened in the phase transition is that when you have your system is around the critical point, you would have this property that when you cross-grain the variables and you make log-log plots of those variables, you would have this scaling variance. So then you see in the second plot that the log-log plot of probability density of a spins in a lattice, you would have this type of linear relation. And then the main idea that was not developed by us, but they developed in the 60s by Kadanoff and then Wilson, is that you can make this systematically, theoretically, in a lattice. And then when you do this, you essentially create a rule to cross-grain your variables. And then with that rule, you substitute your microscopic or more high-resolution variables to a lower-resolution variable. And that the simplest way is just what I say, exchange your up-down variables to the majority. But there are multiple ways to do that. And then if you have a theoretical system, you can have it, you can do it as much as you want because you have infinite lattice. And then if you do that, your system eventually reaches a fixed point. So the structure of this cross-graining procedure stabilizes. And then you can check how your system evolves as you change the scales. And even though this feels very theoretical, it's totally applicable for any like a biological system when you believe that a small change in, let's say, for a disease like Parkinson, a small change in expression of protein, specific protein, can lead you to a disease of Parkinson. So a protein is a molecular level, and having behavior of Parkinson is a behavioral level. So all small change in the concentration of a certain type of protein that's misfolded, it can create a chain reaction across all length scales that change behavior that you have outcome of a disease. So in a way, if you can use these ideas to a real world scenario, which is now feasible because of advent of data, that's a really nice thing to do. And that's what I mean. As a new perspective, how can we make a propagation of signals as a function of cross-graining your variables? Because you would say, oh, I have a signal at the molecular level, how does affect the behavioral level? And that kind of this normalization group was a start for this. And in the recent years, there are developments on this type of theory for networks and complex systems. That's I want to put where we are in this field. And basically, we then did a naive application of this to a population of human brain scans, so 700 people that are healthy, and we make this procedure in a spin, not spin, but time series of rest state MRIs using this idea. And how this idea works in practice, because then I only show theory. So basically, you get an MRI, and then you have to cross-grain the MRI. You say, oh, actually, the highest resolution atlas that is standard by hospitals, like you can do every voxel if you want, but like the highest resolution MRI in the brain is approximately 1,000 brain areas. You could have even lowest resolution, but you got these 1,000 brain areas and you cross-grain. In which sense? You have 1,000 time series, and you have to merge pairwise the most correlated time series. And then I get only half of them, 500 time series. And then I get, oh, I have to merge now the most correlated time series so that I have a lower resolution level. And then I got 250 time series. And I do it until I end up of time series because the brain is finite. And I have also a limited number of data. If we find this behavior I just showed in the previous slide, that in a log-log plot, you have a tendency to be in a straight line, you have a scaling variance also in the brain data. So then, of course, I showed in spin. So in this type of analysis, we also binarize the data. Fortunately, the results are independent of the way we binarize the data. But basically, that's the procedure of this whole context. I can go to the next. So then I have a slide that shows how this would work in the brain. I think that's the next. Yeah, so we choose then a standard health population data set with about 700 subjects that were measured twice. So we tested twice this pipeline. We got a population that is big enough so that we can do this changing the resolution that you see the brain multiple times. Of course, as I say, we already started in very high resolution because in MRI, one voxel has 100,000 brains, or 100,000 neurons, sorry. So it's already high resolution, and you cross grain giving a threshold. Oh, if the activity is high enough, that means that the region is firing. It's not a real firing because it's a bold activity in MRI is metabolism, but has high metabolism. When the activity is low enough, it's not firing. This was also done for neurons from mice. I'm showing what we did for human brains, but in the case of mice, you can do for actual electrophysiological signals, like a spiking of neurons, etc. We also did it for randomized data to see if the brain is randomized. You take like phase randomization of both signals, you will see what this comes. Okay, that's the main idea. So now I show the whole idea in a figure. So you get the brain with 1,000 areas, as you see in the first plot. I only show a few figures, and each one I have a time series. I have then to make this analogy of these spins, and then I say, oh, I need high active and low active during the time of the scan, which is like a 15-minute scan, 2 times 15-minute scan. And then you say, okay, I have this 1,000 binary series. I want to cross-grain then to decrease the resolution of the brain, merge the areas that are active in a more similar fashion. When I do that, I end up with only half of them and do it again and again and again. And then you have this procedure in figures B, C, and D that is just merging all the time series. And then every time you do that, you have a different correlation structure that is a bit more coarse. And at some point, you end out of time series, you get only one time series. That's the merge of all time series. This is finite. With all these caveats, we still try to do these log-log plots that we show in figures E to A. So we compute multiple properties of those time series related to the statistics of the time series. How's the variance of the spiking? How's the fluctuations? How the spectra, like the largest eigenvalue of this correlation matrix that you get when you cross-grain your variables. And for our surprise, otherwise I would not be presenting here, like most of these variables, they have signature of scaling variance. So you get the log-log plot of the data, and you find this. This is not new as a fact of doing these computations in real data. People do it. What is nice is that we had a large population. So we could do this for a full population. And we had a theoretical basis on why we want to do that. So we want to see whether the theoretical results that are known from physics community in normalization could translate to neuroscience. And in which sense? So these numbers, if you compute those exponents in data, you get signatures of scaling variant, but you say, oh, your brain has a fractal exponent of 1.5, and so what? So you have already knowledge that the brain can be fractal depending on how you see the brain. So it's not the point that I want to make here. I want to make the point that every time you compute those numbers, you get a number that relates to fractality of the brain or whatever you mean about scaling variance in the brain. But I want to know whether there is a law or relation between those numbers within a population or even different species of brains. And to do that, you need to do in a full population. And that's how we did it for 700 people. And then if you do it for one person, you get this thing that is already known, not only for brains, but for many, many complex systems that you have scaling variance. And then you get an exponent that is non-trivial, that represents this property of when you zoom in and zoom out, you get similarity. Now then I got to the main result. That's the next figure. If you do this for 1,000 people, what you get, that's why the title of the talk is because of this figure. We computed 3 of these properties can be scaling variant. We compute much more, but these are the ones that we have a theory. So that the others one, okay, let's make a theory by parts. We computed how the variance of the signal activity changes as you cross grain. We compute how the eigenvalue of correlation matrix, the correlations between the spikes change as you change the resolution of the brain. And we will compute how the probability of being silent, because like if you think of binary variables, that's one measurement you can check either you're active or not active, changes as you change the resolution of the brain. So these three numbers, they are scaling invariant. There are way more numbers, but this is the one we could make a theory about what we found. And basically, if you would just compute for one person, you would get a nice known result. Oh yeah, the brain is scaling invariant. This number is 1.4, 1.5. This number is whatever. It's still interesting, but what we found is that when you compute this for a full population, all the points in this curve fall in a single line. And I want to highlight why this line may be important. Each point is a person. So it's not exactly what people call collective intelligence or collective brain stuff. It's not necessarily in this framework, but you see this type of concept. It's like, if you think about scaling variance within an individual, you will just get a number that relates to yourself, how your brain is scaling variant or not. If you get in a full population with the same measurements on their own brains, any measurement that you do about fractality or scaling variance of the brain, in this specific case for time series, they must fall in a straight line because they are constrained by this property of this line. So that is an analogy for the universality that I just mentioned in the beginning of the talk. So in the physics community, there was these universality classes that when you measure these exponents around the phase transition, categories of physical systems would have same classes. So then what we don't call it exactly like that because it's still under review. We're already the second round, but still. But we show that this straight line where all the brain's exponents lie, they can be obtained also theoretically. And if you surrogate the data, as you can see in this figure, there are colorful points. These are the actual population. And then the gray points are randomized population. Even randomized brains, they also fall in this line, but they are just into the corner of this space because when the system are totally independent, you have the exponents must converge to 1. It's a technical detail, but basically when you randomize the system, you make the system fully independent. And then all these exponents that I just mentioned should approach 1. And that's what's happening, but still following this line. And okay, it's beautiful in the sense of mathematics and physics and signal processor strategies to process signal. Why this could matter to the brain? So because this is not random, that means this is an intrinsic signature of your brain. In that measurement, of course, if you measure differently, it can be different measurements. And this variability in this measurement is not random. It's constrained by you should be in that line. But where you are in this line could be correlated to properties of your brain. So variability in your position of this universal, not universal yet, that is like a line where all these exponents lie, must be related to a property of your brain. And then you see in figure A that this variability, so each point in that curve A is one person. And this explains how it relates to the volume of your brain, to the gray matter volume of your brain, which is a proxy for cognition, for age, for lots of things. But so these exponents are related also to the volume of your brain. And as I said, this is also a proxy to cognitive performance. So then human connectome data also has data sets on cognitive performance. And these exponents also relates statistically to the cognitive scores of a PMET24 is a battery of tests you do for assess your cognition. It was straight from like ideas in physics. These correlations are not strong, I may say, but in MRI relations to behavior, the correlations are not strong. Like it's hard to read your mind from an MRI yet. We're not yet there. But statistically, they are significant. So there is a significance relation statistically between these values of these exponents and the gray matter volume of your brain and cognitive tests. Okay, I hope I convinced this is nice. just by the results, like, because the experimental results of a procedure of coarse-grained variables, I would like also to convince that this could be a nice way to go for work with Stefania, that if you think, oh, this actually can be proposed mechanistically, how a procedure of coarse-grained variables is mathematically well-defined, and how signals propagate when you coarse-grained variables, and how you lose signals, because at some point you will lose stuff. Every time you delete one variable, you will have to lose stuff. But how much you lose and how this can be formalized. So I'm happy that Stefania joined a proposal about that we just submitted, because that would be like, in my opinion, one of the edge perspectives in brains, together putting topological signal processing in this framework. Because this is a line, if it's a line, we can try to make a derivation. So we propose a way to derive this line. So I will go now, it's more technical, next slides, but I will just say, okay, how can you derive those lines? So it's a little technical, so please don't, if you can ask in the break. But so the main empirical finds was, okay, the exponents I mentioned to you is first the variance of how the variance changes as you coarse-grain your variables, how the silence of the brain areas change as you coarse-grain your variable, and a spectrometric, like how correlation between areas change in terms of the PCA terms, like the highest eigenvalue change as you change your variables. And this only works for finite systems because brain is finite. The theory we do when you have an infinite system doesn't work, but it's a technical thing, but it's good to inform that. And this works when the system is weakly non-Gaussian, which is the case for the brain. And one reason that the brain could be seen as a weakly non-Gaussian system, like Gaussian plus a little bit, is that the time resolution of MRI is also weak, in the sense that the fast time scales of brain spiking in the brain is about 5 milliseconds. So if you have 5 milliseconds for every spike and you observe a brain with MRI, you lose a lot, because MRI time scales is about 0.7 seconds, 0.72 seconds. So in a classic MRI, of course, there are better machines, they change a little bit, but for every time you will observe a brain via MRI, every data point in MRI, you have in between 2 points, about between 100 and 200 spikes. So you really lose a lot of data when you measure the brain with MRI. It's the best we can have for full brain image, but you have to tell the weakness. If you want to not lose the time resolution, you can go to EEG, to MEG, to other image modalities. But in other image modalities, you lose a lot in the spatial accuracy. So you have to choose where you want to be good, where you want to be bad. So, but in the MRI case, that's what happens. And then the variables are weakly Gaussian. Because of that, we were very straight pointing a way to make a theory for why this could work. Because when the systems are nearly Gaussian, you can use mean field theory, which is a theory that simplifies most of the analysis. So we did the mean field theory on this type of procedure, and we made the procedure in the theory, and then we found this relation with the theory. So that's kind of the view. We also did, because of the referee asked this is not realistic for a brain, because the mean field theory assumes that each part of the brain interacts the same way in average, which is not true, and we agree. So the referee asked to do a non-mean field theory. We also did, but I don't have the slides because it's more technical. In the rebuttal, we also have a theory that is more realistic, where the interactions between the brain are depending on the distance. There's a known in the whole brain in MRI. You can make brain modeling where interactions between regions of the brain depends on the distance between rod A and J, the decay with the distance. So if you also impose the decay interaction at decay with distance, this also holds, but then the derivation is even more complicated. So that's the finding. I go now, before I finish, like to the derivation, but I will be fast. I'll just point to what it is, and if you like, you go to the paper. You can go to the next. Yeah. So basically, you assume this Ising type of model, like interactions between variables, the Jij, and a small field that's in the variable. You can apply a central limit theorem because the system is nearly Gaussian, and then you assume then that the variances are constant and covariance constant. So basically within the same region, the interactions are constant, and across regions they are also constant. There are two parameters in our system, and with these two parameters, this probability, like this energy type of probability of the spin, it's way more simple. That is in the next slide, and then you can express all these numbers I told you analytically. So you can express a covariance matrix for the brain for a mean field type of brain. You can express a variance. You can express the largest eigenvalue because it's a mean field theory. And then when you do that, you know that in an MRI case, because it's a weakly correlated system, the ratio between the covariance and the variance, it goes to zero very slowly. So you don't reach the mean field level in a finite setup. If you would get infinite brain, you would not have this theory. So this theory only works for a finite brain, but it's our case. So our brain is not infinite. It's very big, many neurons, but finite. So with this, you can go to the next. You can then estimate these exponents, how the largest eigenvalue grows, how the variance grows, and then you can get some free energy that's a proxy for the silence of the neurons. And then you just plug the variables, the labels for each exponent, and suddenly you get these relations that are linear relations between the exponents. That's the title of the talk, interdependence. I should put a linear, but it's not linear, will be too big, but linear relations between scaling exponents in the brain. Yeah. Then this line is kind of very, like, that's why the R square of this line is 0.98. The actual theory tells that round these numbers, then you have the theoretical result. And that then from this mean field derivation, we would get this result. It's kind of, how can I say, we didn't explore fully the consequences for brain itself. We focused, like in the theory, it was more mechanistic, like, okay, if the brain interacts this way, you cross grain that way, you find its exponents, and they are in a line. This could be exploring a different perspective, more, okay, what this would happen if you do this analysis in a health versus a degenerative disease, or other types of setups. So on the tasks that we didn't explore this, we focus on, okay, this law exists. We cannot claim it's universal law. It's under the constraint we made of MRI measurements, weak correlation Gaussian variables. We made a theory, and then and it's a complex system that has this type of exponent should fall in that law, in that line, because the theory we made, as you saw, was not related to the brain. It was related to spin variables that you cross grain those variables. So the brain doing the theory was just an inspiration. So we got, okay, there are spin variables. I measure the variables as we did in the previous with the theory. And then if you do this in this way, you get this line. Yeah, that was kind of, I want to point at, because this is open a perspective. Okay, if this is good, what can you do with that for the brain? That one direction. So I have colleagues, I worked in the hospital before, moving to the math department, that they are trying to do this in different image modalities, and for like Alzheimer's patients, or these different setups. We have an idea, but it's not yet proven, is that if you see this line, as I show you, randomized points are in these gray points. So if you think when your brain ages, it gets a little bit more random, or when your brain gets a degenerative disease like Alzheimer's, you get a weak correlation, you should go down the line. But for that type of disease, in another types like epilepsy, where you get over synchronization, your correlation gets strong, you should go up the line. But that's not clear what is happening, because it's in perspective. Okay, I think we are close to the end because of the timing. But the discussion is that, yeah, we found the somewhat scale structure of this exponents within a population level. These exponents correlate to cognitive performance. It's really related to critical phenomena, as I showed in the beginning. This was made theoretically for a time series that are weakly correlated. It was not narrowing the brain in the theory. So this could happen in other systems where I have weakly multivariate correlations. And we'd like to see, we can go to what comes next. So What comes next, the experimental brain side is test this in different setups, different brain modalities, different conditions, a task or disease. And the theoretical setup, that's why I'm happy to share this with Stefania and Breno, is to make this in a different approach for renormalization, how to cross-grain variables within a theoretical setup, let's say we do a TSP, and the moment you do a TSP, you start losing the vertices. You merge the vertices. There's one extra thing that happens during a TSP or even during a graph signal process. I am aware, not my field, but possibly wavelets or other types of techniques could also address these. But in a way, if you want to follow this type of cross-graining, it would be how a signal survives when you measure variables in a signal, and why this is relevant, because this is actually happening in our body, like from cells to behavior. Then it would be an inspiration to come with new cutting-edge techniques. And is an experimental, like a motivation. A theoretical motivation comes like from physics, from these normalizations I told you, and the preliminary results, not only from myself, but from other groups that are doing theories on this direction on networks, like groups in Italy, in Netherlands, across the world, that this could be a good perspective. I think that's it. Thanks so much. Sorry I spoke too much.
Thanks, Fernando. Thanks. Okay, we can start the next talk. I will introduce Professor Alessandro Crimi. He's a biomedical engineering and health economist, both career bridge neuroimaging research and healthcare management in low-income countries. Born in Palermo, he earned an engineering degree from the University of Palermo, a PhD in machine learning for medical imaging from the University of Copenhagen, and an MDA in healthcare management from the University of Basel. He has had a research position at INRIA, ETH Zurich, IIT, and the University Hospital of Zurich, while also teaching and leading a healthcare project in Ghana and other sub-Saharan countries. For 8 years, he was a lecturer at African Institute for Mathematical Science. He is currently professor at AGH University of Krakow, where he coordinates the course Machine Learning for Neuroimaging and Neuroscience. He will hold the talk from causality to graph attention networks for brain diseases. Thanks, Alessandro.
Good morning. I hope you are hearing me. And thank you for the invitation. I would like to be... I hear myself like a weird echo. I don't know what to... Oh, now do you hear me? Ah, okay. I was from your microphone. Okay. Yes, so I would like to be there. Instead, I'm here in the cold. I was bugging in the fog today. So if it's okay for you, I share my screen. Okay. Which, given the people there, I made the small change. Do you see it? So I changed a bit. Instead of just a graph attention network, maybe something more related to topological data analysis that we also did it. Can you still see it? Because I don't see your faces anymore. Maybe someone can say yes. Yes.
Yes, yes.
Okay, then I continue. Yeah, so as I will, yeah, but I hear myself again from the microphone. Yeah, the original plan was to talk about effective connectivity and then some graph attention network, which I made some changes. Like I will just mention super quickly, and instead I will talk about something about epidemic spreading on graph and hypergraphs, because I guess will be more relatable to the other talks today. So I go super quickly because I think everybody there know what's structural connectivity, so that we kind of given some region of interest, we represent the physical connections within the brain. Functional connectivity is mostly related to co-activation of the different brain region, while the effective connectivity, which is still kind of highly debated, what is it and what does it mean and what represents, generally it's referred to the fact that we have some activity in some brain regions, and after I'm hearing myself again. Some activities in some brain regions that afterwards will cause some other activity in another brain region. So there is kind of a cause and effect relationship, which of course is mirrored by some physical connection that they probably have to exist, and also some functional connectivity that must exist. So it can be seen something like, at least I see it as a combination of this, and can be used either to find some biomarker for disease or for studying some internal mechanism of the brain. First of all, there are a lot of debates what causality means, and just to give you a reminder how difficult it is to relate causes, there was the famous paper about chocolate causing a Nobel Prize, where they were showing that that there was a strong correlation between chocolate consumption per kilogram per capita in a country and the presence of Nobel Prizes in that country. So this is to point out that effective connectivity is not functional connectivity, because obviously it's not enough that you eat chocolate and you win the Nobel Prize. Instead, what's mostly likely to happen is the fact that there are some countries with a high domestic product, so therefore people are spending more money in everything, including chocolate, and also they can attract good talents. So there are people that end up having a Nobel Prize. So these are kind of spurious correlation that we see, and kind of this, in the brain we might see similar things. There's been some models and tools that have been proposed. Some are more friendly, some are more heavily computationally, some have been debated that they are just verifying but not helping causal discovery. So it's still an open world full of discussions. What I start using it, not because I believe actually it's one of the most criticized methods, but it's actually the easiest one. The simplest computationally that you can use in large scale networks is the Granger causality. Long story short, imagine you have two time series, and we see that, for example, changes in one time series after some time cause some changes in the other time series. We might be prone to say that X is causing Y. Obviously, it's not just a test of done initially with some kind of autoregressive coefficient, but also it's verified afterwards with some statistical test if the prediction of the series itself it's improving if we combine with another series. It's, as I was saying, is highly criticized, but it's the easiest to implement and the one that can scale easily, and is the one that I was starting investigating. And the idea is that we don't consider then a connectome just with the edges, but we also put some arrows in the edges. One thing that I started doing some time ago was to kind of define this effective connectivity not just in the traditional sense of Granger causality, where we look for the coefficient for this Granger causality, but also kind of put in some kind of constraint related to the structure. So it was kind of a way to put functional and structural together, in the sense that generally these terms you see x of t and t with some kind of delay is from functional data, and generally the matrix A is kind of free. So instead we found a way that we can kind of constrain the matrix B to always reflect structures, kind of putting together function and structures together, which is something that you will see often in proteomics, that they try to merge some data related to a shape of a protein and its structure in 3D. Okay, I go now. Initially this was tested on some autism spectrum disorder disease, and what we noticed that was that first we noticed that the classification between typically developing children and children with autism spectrum disorder was better with the effective connectivity. So in this picture you see the first column, the features that we found using SVM and some structural data. In the middle are the features that we were used by SVM support vector machine for functional data, and in the last column what we have seen that were related to the effective connectivity found with our constraint method. And apart the fact that the classification was better, what we noticed was that kind of the effective connectivity was kind of mixing the two representations. So it was still strongly similar to the features that they were used for the functional connectivity, but they were also kind of including some that we were seeing in structural connectivity. So kind of telling us that it's kind of a way to put together function and structure. So this was the original work that we did it. So then we continue in this kind of universe. I don't explain you what's a SUGIAR because we don't have time, but there are other ways to consider this kind of effective connectivity, starting considering, starting seeing it as a dynamical system. Actually, maybe I have time to show you a small video. So what this Sugihara means, apart the fact that they change a bit the perspective of how we define causality, is like to show you that if we have, for example, this 3-time series, that they can kind of represent a dynamical system on its own. So it's not just looking at some linear correlation, linear relationship of autoregressor for causality. But imagine you have a first-time series x of t, then another one, which is simply the other one, the original one a bit delayed, and the third-time series that is the other one delayed. So these are for sure related because they are the same signal, just a bit delayed. If then we look at it, at the three features together, of course, this is kind of an ideal case, so that I am representing these three together. So we might make them in a way that can resemble the dynamical system that representing some kind of attractor, in this case, like the Lorentz attractor. The whole story of this is that then, since it's represented in this way, we maybe start doing other stuff like looking at shadow manifold and all these things to kind of relating, and because shadow manifolds can also be separated according to variables, so we can start looking if there is a causality in this. This was actually just kind of an introduction to what I'm going to tell you, because then after we had this, we start using some more fancier approach related to machine learning with something called reservoir computing, because it's something that is kind of more adapted to represent some dynamical system, we noticed. For those who never heard what's reservoir computing, the idea of reservoir computing is that we have some networks which are randomly connected, and these are not optimized. So what we try to optimize is kind of the initial layer of some weights that allow us to project the input time series into the reservoir computing, and then also we have some other another layer of some weights that are projecting back to the original representation of the time series. The only thing that we, in a traditional sense of machine learning, is optimized is this the last layer that is allowing us to do the representation. And then in the end, this is kind of used similar to, like, it's not that far from Granger. We can use see it as a Granger. We use it more in the context of Sugihara causality. But the idea is that then if you are able to predict a series better, you can kind of relate this to some kind of causal relationship. The ins, of course, then there is all these discussions again. Is this real causality? Is this temporal correlation? But anyway, in our context, we were more interested to see if this representation could lead to better reconstruction of the better prediction of the signal. And this is what we observed. Why is this better? Okay, first of all, because traditional Granger causality is linear, so this kind of approach is for sure creating some kind of non-linear representation, and is for sure going to a higher dimension. So it's like in the old school method when we were talking of kernel PCA that we are projecting in a higher space. So this is what is happening. And yeah, we also put it together in a larger framework related to stroke. So what we were interested to see was to kind of observe if stroke is causing for sure some damage, both structural and functional. And our idea is that not only causing this, so this actual damage of stroke can impair also miscommunication between brain region. So we attempted to see the stroke as an effective connectivity disease, substantially, because it's not just that there is a damage, but or in at the structural sense, so that stroke is damaging something in the brain, but it also kind of interrupting some communication. So it enters in the world of damaging the level of transfer entropy from one brain region to another one, signal communication within one part of the brain and another one. So what we did practically was that we had some patients, healthy subjects, and also patients, and we put through we extracted time series according to an atlas, and we try to see if we can estimate the effective connectivity using our representation with the graph with the reservoir computing, and afterwards we fed this entire representation into some kind of graph convolutional networks to apply some kind of classification. So the idea was not just to classify healthy subjects from people from people having a stroke, because obviously if it's enough that you look at a standard MRI to see that someone has a stroke. What we were interested was more like extracting the brain regions that were doing more in the context of attention networks and explainability, trying to figure out which part was more representative in this classification, because substantially it was also the part that was kind of highlighting as eventual miscommunication within the brain, and possibly, of course, not looking at the specific case of stroke, but trying to see if there is an overall network disruption caused by stroke, which was challenging because obviously each stroke is a case on its own. They tend to be more or less close to the Willis circles, but it's an heterogeneous disease, so it's kind of very difficult to find some kind of general biomarker that can highlight some miscommunication within the brain. But we kind of found it. And what was also interesting was that I don't know if you can see, maybe this picture is very small. We tried to look at if there were specific difference, at least from the side of the stroke. And this is what also we noticed, that there was more pronounced damage, that there were more misconnections depending on the side of where the stroke was occurring. So it's not just the size of the stroke or the intensity of the stroke, but there is an impact on which side of the brain is happening. And this is probably mostly related to the fact that there is a lateralization in the language. So some people have more developed the language network in one side of the brain. So if the damage is happening in that side, it's more difficult to the brain to recover generally, not just of the size of the brain, but of the stroke, but on the fact that the stroke is happening in the same side where they have the most developed language thing. And yeah, so then this was also, I don't go too much into this. So we wanted to see if it was not just related to the effective connectivity that we have constructed. So we run more or less the same study of lateralization using brain gradients, which are not something related directly to connectivity, but it's more like measuring the difference of action connectivity between one brain region and another one, not at a region of interest, but more like almost at voxel level. And we found like the same thing. So we also noticed that there is this impact of difference in lateralization of left and right, depending where is the damage. This, of course, this was all theoretical. Like we kind of pointed out what was going on, but it was mostly related to the fact that we maybe also this miscommunication as we can highlight. This is one better. Maybe some miscommunication we can highlight. This can be like an insight if someone wants to do some therapy like stem cells, that maybe they can pilot more on specific connections rather than attempting to spread stem cell all over the brain. So because we can kind of highlight which miscommunication is affected within the brain. So I shared some stuff that we just submitted recently, that more we enter in the realm of foundation model, and we investigated a bit more how if we can use some of these things. So because after the explosion of ChatGPT, everybody's building foundation model for everything, and in fact there are also foundation model for time series. Some have been already published, some are in development. We also try to build one or to use the previous one. And the general idea is that we, instead of using foundation models to predict words, to build sentences, like you can see with ChatGPT, that you ask a question and there is like a series of words, one after the other one. You can imagine this foundation model as something that they can predict how a time series will be. And they follow the same principle. So there is a tokenization, there is a transformer architecture like for ChatGPT and so on. Of all the methods we kind of study or try to build, we found the most interesting was one called times FM. And the reason why it was interesting for us was because it was allowing to make time series forecast using either the series itself or including more series for as a covariates. Why we were doing this? Because we attempted to kind of reconstruct the Granger causality approach. I don't know if you remember that I mentioned before. One way that we can see is that in Granger causality, if you can reconstruct a time series just with the series itself, or combining to see if one series is helping the prediction. So this is how we kind of twisted this foundation model to see if the foundation model is able to predict the time series on its own, or if it's using another variable. And in that case, we might speculate this kind of Granger-like prediction. So if it's able to increase the prediction statistically significant, it means that they also it is sort of also kind of Granger causality obtained with these models. What we noticed was that first of all, for the stroke patients, we also use the same subjects, and with for the previous study related to stroke and this classification, we did it, and we haven't found a special particular difference related to if we were using together all the time series. Also, an important thing, we were using these models as a zero shot, so the models were not further training. So these were models that they were being built maybe from econometrics time series, and we just used them to see if they were working, while instead the Granger causality is still related to autoregressors that have been trained. More or less, we got at the same point for overall subjects. If we were using only the patients, the model, the time foundation time series model was predicting better. So it seems that was more prone to strange signal or disturbed signal. And also since of course it's kind of difficult to, so this is just a prediction, these were kind of difficult to evaluate that this was a real causality, because of course we used previously as to define effective connectivity for a classification, but it was not, there was no ground truth. So it was us looking at the stroke to see if there was some kind of disconnection. So it was kind of defined by us, the effective connectiveness. We tried it to study it like from the point of view of synthetic networks, some networks that were maybe built from few nodes with logistic map coupling, or something more advanced with the Ornstein-Uhlenbeck process, where we know the ground truth, so we could check if the causality that was detected with the foundation model, it was more precise than Granger. And it was interesting that it was dependent on the density. So depending how many causal relationships there were in the network, either there was not much difference, or was sometimes even Granger was better, Granger causality was better. But if we were increasing the number of causal connections within the network, the foundation model will start being more accurate. So there was more precision, and also the recall index was better, and also the general trend was better. So it seems that Granger causality tends to be more conservative. They tend to miss some to find some connectivity, while the model, the foundation model, because it's trained probably again with vision transformer, which are probably finding some nonlinearity, was more accurate, was tended to be more accurate. Then I do some kind of some fonts disappeared. I do some unashamed advertising. So we try to pack all these things I show you about effective connectivity in a general toolbox. Both that it's everybody wants to, you can download this freely, and also comment open issue if something is not working. So we put everything, some Granger causality, more foundation model, transfer entropy, and everything, so that can be accessed. And this was because many of these Granger causality models we found, they were mostly in MATLAB. We never found anything reliable in Python, so we decided to build it. And also we had some kind of visualization, because another thing that we found was that is kind of problematic each time to, it is not starting. It's kind of problematic to start to visualize directed connectivity. We didn't find any proper tool that was allowing us to represent the arrows that I was telling you, maybe navigate on it. And also in this tool, you can find also that dynamically, you can also load some tractography, and the color of the directionality is changing. You don't see this because you will need a video, but like the shades of white is moving toward the center to highlight the fact that there is a directionality from here to here. So if someone wants to take a look and try if there are some bugs or a proposal, please download it or open some issues. Now, if it's assuming I didn't go too fast, I move on the other topic, which may be more related to the people in the room. Are you still with me?
Yes, yes.
Okay, as I say, I kind of reduced the story of graph convolutional networks, but I would like to talk about something related to Alzheimer, and not that in the end will lead us to kind of topological data analysis. And the idea that we have been investigating was related to the fact that we can see the structural connectivity not as a feature on its own, not that we start looking. Of course, you can also consider this for neurodegeneration, but the idea is that some neurodegeneration, they use the connectome as a highway, But instead, what's more happening is that from the edges, from the nodes to other edges, there is more like a spreading of misfolded proteins. For example, in some neurodegenerative, in kind of all the neurodegenerative disease, there is a protein that is spreading and causing some damage. So this is more from amyotrophic lateral sclerosis to Alzheimer to frontotemporal dementia to Parkinson. There is always a protein that is kind of spreading and causing some damage and ultimately causing the death of some neurons, and then of course some disability and all other stuff. From the point of view of Alzheimer, there are two main proteins that are involved in this. So we call them one amyloid beta and the other one is tau filament. And generally what's happening is that the first one starts spreading is the amyloid beta, where we have some kind of phase of not so much cognitive damage, but they're still having some kind of mild cognitive impairment. But afterwards, after some years, that there is the spread of this amyloid beta, for example, from one brain region, it starts spreading all over. There is the second one. There's the second protein that is starts spreading. That's the tau filament that is causing more damage. So we have more. There is more relationship with on the spread of tau filament and disability we see in subjects. So we start seeing that they're having more serious disability, more cognitive impairment, more memory loss, and ultimately, like then they die substantially. So this is the real culprit, but the first protein is the one like paving the way. So the kind of study we did was like given that we have some structural connectome and some initial deposition of either amyloid beta or tau filament, if we can predict how will be the spread afterwards, because this might be useful to kind of say how fast the subject will degenerate, or even so maybe giving some kind of insight to some doctors. Currently, there has been tons of approaches for Alzheimer's disease, but nothing really works. So it's kind of still lost battle, but still, if we can have some aid some doctors with this kind of prediction, we still believe it's kind of useful. And in the initial stage, what we did was to take the same approach we were using for effective connectivity, but not looking for effective causality, but more looking if we can predict future deposition. In the same way we used to see if a time series can predict another time series, we were looking if one deposition of protein can predict further deposition of proteins. So what's kind of similar stealing from Genscher causality. And also we start looking at directly to graph convolutional networks if in the same way, if some kind of input features of protein deposition can predict future deposition. Looking at the state of art, before we attempted these two things, there are two other methods that are considered the most in literature. You will find that they are the state of the art, and one is the famous epidemic spreading model, which was literally considering the spread as a simulation, and another method that is called network dynamics model, which is kind of inspired by it equation, also more like in the context of simulation of spreading of considering instead of changes of temperature, they were considering the spreading of these proteins here. In the initial paper of Luca Gherardini, he investigated just the normal connectome. What we found, okay, I don't go too much into how you do the message spreading of graph convolutional networks. What we notice, so here we have the representation of all the region of interest and the prediction that you can have after some years using all these four methods. So the autoregressive model way to predict the spreading, the graphical convolutional networks, and the two state of art. What we noticed that generally for this type of problem, actually graphical convolutional networks was the worst in some cases. So here you see the error was higher than the others. And generally the autoregressor approach kind of, because it's a data-driven approach more in the context of on the direction of machine learning rather than simulation, was kind of performing generally better. That does not mean that then the second winner was this epidemic spreading. So this was, we were seeing better results, but the epidemic spreading was not that far, so was also kind of very similar. In fact, in some cases we did some statistical testing, statistical significance. In some cases there was no difference. In some cases our proposed method with autoregressor was better, but in some cases the other method was still good. But still, even if they are comparable, we can discuss the fact that we have an alternative way which does not require simulation. And this was in 2023. So what we learned was essentially that this the MAR model, so it's a data-driven approach that can be help us. It was kind of copy inspired by effective connectivity and was kind of obtaining comparable results. So more recently, I had a student. There was a student that was considering also some other approaches that was introduced by Jacopo Jacopini and Vito Latora that were related to the idea that maybe the spreading is not just a binary relationship. So it's not that, of course, if you have a node, of course, you consider the degree and the entrance in this kind of network representation, but maybe we should consider something more related to higher order than just binary connections between within the networks. So through the starting from old works from Vito Latora and Jacopo Jacopini, we investigated if there is a way starting with this kind of contagion within some kind of high order representation. But the idea was kind of similar. So we had some baseline protein, and we had now a connectome that was used to create some high order representation. We went only to the first and second. We checked the second order because it was start being computationally heavy to measure this. And we were using some kind of spreading model initially to see if was allowing us to have better prediction. So the idea was that we kind of consider everything as a microscopic Markov chain, where, of course, at some point there is a coefficient of probability that is relating the entire simplicial complex, not just the binary connections relationship between the edges. And what we noticed was that the simplicial complex was kind of beating the one that we thought before was the best model. So if it was beating the best one, means it was beating also all the others. What was more interesting is that there were further improvement depending on something related to what I mentioned you before, that there are two proteins, and the fact that also one protein is more the one that is causing the real damage, and one is the initial protein that start doing some mild cognitive impairment. Why this was interesting? Because it seems that at some point the one that is not causing a lot of damage but it's paving the way to the other one, at some point has kind of plateau. So when the second protein start making damage, the first one is kind of not increasing that much. Why is this annoying? Because the other methods were having some problems to make prediction in this part. So they were more or less continuing to make prediction that were exponential for the first protein, while the simplicial complex was kind of handling better this situation. So we had less reconstruction error from the position of proteins in these cases. Obviously, this was the things that we had published at the latest EMBC, that the simplicial complex was kind of beating the other methods. We had some preliminary results using hypergraph convolutional networks, comparing graph convolutional networks. And we more or less realized that we have been comparing apples and oranges, in the sense that this was a simulation method. Of course, it's true that it was beating the other method, but should be more comparing the simplicial contagion model to the original epidemic spreading, because this is more like an epidemic spreading on a normal connectome, while the simplicial contagion model is something that is happening in higher order. And anyway, in this case, they are. It was better. So we noticed that there is some kind of nonlinearity or advantage using higher order networks than using just a normal epidemic spreading. Instead, if we were using, I don't have the results here because the students finished this thesis and now is doing another job. I don't even know if this work will ever finish. What was noticed that higher order graph convolutional networks was even performing better than everything. So maybe the high order graph convolutional networks is not maybe what we noticed is even beating the simplicial contagion model. So anyway, we are not done with this. So these are still kind of preliminary results, but it seems that so far the best of all, if you want to look at prediction, is high order graph convolutional networks. I stop here, and I know I was promising some other stuff related to graph convolutional networks, but I think this has been already a lot, and probably people have also lots of questions, complaints, concern about everything I said so far. So I don't want to add the third topic, but if you want, we also have done some stuff related to graph convolutional networks to make some kind of classification. What I would like to close here, so summarizing what I talked so far. So we have been interested mostly in dynamic things. So in trying to see how one part of the brain is causing some effect in another one, that is called this affective connectivity, which I mentioned even myself, that even the methods we have developed has probably some clear limitation, like that you can argue that what is real causality. So we still might risk to be in the story I mentioned before about the chocolate at the Nobel Prize. So all these results have to be taken with some pinch of salt. And also the second part of the talk, we noticed that we can have better prediction of misfolded protein spreading if we use high order interactions. And maybe the future trend will be that we also add some further things within this kind of overall story. So maybe we can also consider other aspects of high order interaction and effective connectivity, still keeping this directionality and all the other things. With this, I close because I think I should allow at least 15 minutes to questions or discussions. And if you want to reach me, this is my email, or feel free to reach me also in other kind of social media. So where is it? I stop the share or screen sharing, or if you want, I'm here for you.
Okay, thanks. Thanks very much, Alessandro, for your very interesting talk. There are some questions. No, there are no questions at the moment, but
they were waiting. The graph convolutional networks was a bad idea to change.
It is interesting also to take into account of the direction of the graph. Maybe for simplicial complex, taking account of the direction is more hard task, but anyway, it is very interesting. It is very interesting. Okay. Thank you. We keep in touch. Okay. And
Okay, thank you for the invitation.
thank you. And we can proceed with the next speaker. The next speaker is Professor Luca Mesin. He received the graduate degree in electronics engineering and the PhD degree in applied mathematics from the Politecnico di Torino, Italy, in 1999 and 2003. He is an associate professor of biomedical engineering and the supervisor of the mathematical biology and physiology group, Department of Electronics and Telecommunication, Politecnico di Torino. His research interests include biomedical image and signal processing and mathematical modeling. And then the talk is a joint talk with Dr. Hossein Ahmadi. When I introduce him after, or okay. He received the Bachelor Science degree in Electronic Engineering from Kurdistan University, Sanandaj, Iran, in 2010, and the Master Science degree in Telecommunication Engineering from the Amir Kabir University of Technology, Tehran, Iran, in 2016. Is currently pursued the PhD degree with the Politecnico di Torino, Italy, specializing in electrical, electronics, and communication engineering. His research interests include brain-to-brain communication, semantic communication, and signal processing. Okay.
Okay. Thank you for the presentation. Thank you for inviting us to join with this very interesting workshop. I will talk about EEG for developing brain-computer interfaces. I will talk about mainly about problems, and Hossein will present some solutions that we are developing and which are very promising now. First of all, EEG is problematic as a signal because it reflects the activity of a very complex system, and it is a signal which is also noisy. So, first of all, why is it so complex? Because we have so many cells in our brain. There are a lot of glial cells, but we are mainly interested into neurons, and we have 80 billions of neurons, and we have 10 to the power 15 synapses, and EEG is related to postsynaptic potentials mainly, and so we recognize that there is really a huge number of sources for our signal, and so for sure there is also great phase cancellation, information loss, and this is one of the reasons why it is quite difficult to study it for a few channels, in the sense that even if we have a high density acquisition system with 100 electrodes, 100 is really a very small number with respect to the huge number of sources that we have. About noise, well, there is a lot to say. I simply show you an example of EEG in which there are some artifacts, for example, blink artifacts, muscle artifacts, even very far sources have a large effect in terms of amplitude, in terms of power into the EEG that we are recording. Now, we want to summarize the information included in the EEG using some feature. And so why? Because EEG has a low signal to noise ratio. There are artifacts. There is a volume conductor problem, as each source is connected to provide an effect on different electrodes, and it this effect is affected by the volume conduction in between. It is a non-stationary signal, so it is a very difficult signal, and finding robust features could be a good solution. As the features can compress the information, and if they are informative about what we need to interpret brain activity, this could be a great goal which is achieved. What should the features do? They should denoise the signal. They should be invariant in some sense, which means stable across different recordings, for example. There is a huge collection of features that we can use. Some ones coming from the time domain, frequency domain, time frequency, time space, connectivity, so an important field in this workshop for sure. Non-linear features reflecting complexity, fractals, so what we have been hearing in one of the lectures today. There are also some differential geometry features which can be useful. But all these features provide some information, condensed into a scalar and vector matrices. But which feature should be chosen? How can we select the feature? How this feature match the model that we use to interpret our data? So this is a great problem. We can use these features within the framework of classical machine learning, for example, in order to try to map our data into some intention, for example, some classes among which we want to choose in order to interpret what is thinking the user. So in classical machine learning, we don't use raw EEG, but some features extracted from it. We could say that there is no universal optimal pair features model in order to solve our problem. Even using cross-validation, we can get very good performances, but usually these performances are only specific for the data set that we are using, and so this cannot prove optimality, since by changing the acquisition system, changing the day, taking out, removing the acquisition system and replacing it, we could lose the accuracy of our model. So the consequence of not having a way to select a good feature and of producing a good feature is that we have a weak transfer across different paradigms and different tasks, etc. The main problem maybe is that features are restricted by predefined recipes, and these are not adapted to the specific user, and so maybe this is one of the reasons why those features sometimes are not highly performant. About the deep learning extract automatically the features, but the architectures that we select matters, in the sense that there are some architectures which favor spatial distributions, and so, for example, CNN. There are other architectures which favor the temporal dynamics, RNN, for example, transformers. And when we try to put together CNN, RNN, trying to get the benefits from the two architectures, usually we are not able to find a great improvement. So this is what we noticed during our activity. So single branch models are focused on specific space or time specific dynamics, and it is not so easy to couple them in order to get both domain represented. So this is a core challenge in our opinion. Try to capture the time-space dynamics, avoiding overfitting and trying to get good results, even without requiring a massive amount of data, which is usual when you are using deep learning models. What is our objective? What do we want from a BCI? We want that it is robust within or between sections, subjects. We would like to get a calibration in short times. We would like to be able to transfer from the lab to the to practical application with minimal retuning. And we would like also to be compact, to have care of the privacy, because we would also like to transmit our information across different systems. And it would be nice if we could finally get a simple decoder, because this would reflect also the stability of our approach. This is an example of results that we obtained. Very good accuracy in sections, but applying the same method the next day or recording the data with a new device, accuracy was not the same. So how can we solve the problems? We think that there are three main approaches. One is trying to fine-tuning the classical methods, try to get better features, smarter calibration. What else? Trying to learn a real model merging space and time properties, or trying to change our perspective, maybe trying to focus on semantic information, which is what Hossein will talk about. Once we get our results, how can we say that our brain-computer interface is good? We can make different kind of tests. One of the tests that we prefer is that leaving one subject out, because in this case we could ensure, we could try to support that our brain-computer interface could be trained in our company, in our lab, and that could be useful outside with minimal retuning. We could use many metrics that you know. We can try to stress our model by using more artifacts and noisy signals, trying to use a few data to train, trying to reduce the resources and see what if our method is still effective. Then I can leave to Hossein, who will introduce our methods, different methods, which are proposing a different perspective and which are obtaining promising results. So we want to go on in this direction. So thank you. Maybe Hossein can come here and we can go to the second presentation.
Thanks, Luca.
Thank you, Professor Calderetti, for inviting us. Thank you, Professor Messin, for opening the topic. And now I'm going to introduce some solutions that we have for the problem that Professor Messin introduced, the problem of finding the optimal feature for brain-computer interface, specifically for EEG. And then after introducing our solutions, briefly, I will discuss about the application, few examples. Let's first of all introduce the end-to-end flow, and then we will go to
the solutions. A non-verbal individual has several physiological needs like everybody, but the problem is that how can express those needs. For example, I need some water. I want to go to the bathroom. I want some coffee. I want fresh air, some fresh air. I want to go to the yard. There are several physiological needs, but the problem is that how to understand what they need really. So we can record their EEG signals and pre-process their raw EEG data. But the problem starts from this point. Now, which feature can help us to understand what they want? There are several traditional features, for example, entropy, PSD, several others. But which of them can help us to translate those intentions, what they want, what they think actually? This is the first problem. We have a solution named semantic feature extraction. Later, we will discuss about this in detail. And after that, the second problem will start. How to send those intentions, those translated thought, safe and securely to the point that we want. For example, a nurse, a doctor, a caregiver. So this is the second problem. We need a wireless link, but it should be safe and secure because it's about privacy. It's not about something less important. We have to secure the path to save the information. And after that, we can decode the intent that we have received here by the caregiver. That decoded intention triggers a signal for assistive action, and finally the result will deliver to the subject, the user. So as I said, there are several problems from EEG acquisition, pre-processing, semantic feature extraction. Here, by semantic, we mean the intention, the meaning behind what the subject thinks about, okay? And then safe and securely sending those translated intention to the destination, either a caregiver, a nurse, a doctor. And this is only an example. There are several usage later we will discuss about it. So here is the end-to-end flow. What have we done in this regard? For the problem of selecting the optimal feature, this is our first paper introduced and presented in EMBC conference in July, 2 months ago. It is simple but nice, honestly. Today, I don't want to go to discuss about it. It has two phases. This phase is important. I will go to discuss about this in detail. But this is something we can change it. While here we have 5 convolutional neural network blocks, but we can change whatever you want. We have tried several different, even simple machine learning models can give us almost the same result. But what is important, this first phase, which is aligned with the challenge, the problem that you write. What is that? Okay, here is a single trial for real EEG data. Here, we have only 5 channels, but the data actually has 124 channels. Now, the problem is that which feature can help us to understand what is this? We have 4 seconds, but how to understand what is this actually? Okay, we have spatial and temporal features, but do you want to use entropy, PSD, connectivity features? Which feature can help us? What is this? Okay, there are several candidates, and we have several papers use all of these features. For some cases, we have the result near perfect, more than 99% accuracy. With the same model, when we go to another data set with the same task, the same paradigm, we see a drastic reduction in performance. What is the problem? What is the hidden point that we don't understand really? And it's not only for our work, we see in almost every other models in BCA domain. Also for transfer learning, there are several top models like EEGNet, Conformer. We have almost tested all of them. For some cases, some paradigms, some data sets, we get very good results, but for some other, no. And there is no answer why. Because machine learning and deep learning models are black box, and we don't know really what happens inside them. And maybe also brain is black box. So we have two challenges. So our first solution, which we can use it in any case, for any test, any paradigm, is that think about how we can extract both spatial and temporal features. If we convert this wave-based EEG into a topographical map, for example, why doesn't work? Okay, a topographical map. Here, we have all 124 channels, but we lost the time. All 4 seconds now is shown in an image. Now, while we have captured the spatial aspect, but we lost the time. Okay, this is a great idea. Let's go continue and split the same 4 seconds into more segments. For example, here we have 5 segments, each of them 80 milliseconds. And in this case, we have 4 topographical maps. So instead of 1 image, now we have 5. The same spatial aspect, but more temporal resolution. Then if we continue in this splitting, maybe we get more. For example, the same 4 seconds, if splitting into 20 milliseconds for each segment, we get 200 maps. This is a nice solution for data augmentation. One of the most important points in almost every area of data science for deep learning and model and machine learning is data augmentation, increasing data artificially. But data augmentation is not valid for biomedical data because EEG is unique. We cannot increase EEG but by a common data augmentation approach. But now, without any artificial data, we increased the samples. We have still the same 4-second length, but in each of them, even with 20 milliseconds, the topo maps are completely different. So instead of only 1 sample, now we have 200 samples. So we can train the model very well without data augmentation. And we captured both spatial and temporal aspects of the data. This is like we have a video and record what happens inside the brain. What is better than this? What is the purpose of feature extraction? We want to find the feature is something that we want to use to help us to understand what happens inside the brain. So what is better than that if we can have a video to record inside the data? This approach is exactly like that. It seems we have a video and record the inside of brain. while still we lose some temporal aspect, but now it is about 20 milliseconds. Even we can reduce that that segment, but for our test, 20 milliseconds is almost near optimal, and we have obtained very good result our several different data sets. So in this case, we can transform the raw EEG data, the wave-based EEG data, into the sequences that can capture both spatial and temporal features. And each topomap, each image that we show has a unique representation of that time slot. And this is very good for some deep learning models like CNN, which is image-based models. The problem for deep learning models, Professor Messin mentioned, is a mismatch between the traditional features and those models. For example, for CNN, which is initially designed for image-based data, we can use for EEG for capturing spatial features, but it is not good for temporal features. From other side, some other models like transformer, like RNN, LSTM are perfect for temporal features, but we lost spatial features. But now each image, every image has both temporal and spatial features. This is the first solution. We have used this model, our several different data sets, but later we will discuss about the result. Let's introduce the second approach that we have. It is more universal. And what we mean by universal, when we use universal, it means this case, it is not only about temporal and spatial features. It is about the task. As you saw in the first slide, we discussed about intention decoding. It is not only simple motor imagery, task like left hand, right hand. Please notice, I'm saying simple motor imagery task. Motor imagery is not simple. Actually, it is very complex. But compared to what tasks we use, it is super simple. Yes, motor imagery is very complex compared to ERP, event-related potential tasks, which has two cases, target, non-target. But when we have a complex task like, I want some water, it is not a concept. So motor imagery, which is a complex paradigm, compared to those tests that we use in real time, and later we will show it, it is very simple. So we need a universal approach that even can decode those complex tasks. Here is what we introduced in neural engineering a few months ago, published. The detail of the approach, the criterion for semantic. What is semantic? Is semantic initially introduced in NLP, natural language processing. But what we mean here by semantic is quite different. Semantic in natural language domain is about the different meaning each word has in a sentence. But now, by semantic, exactly we mean intention behind the thought. Okay, so, and the criterion for defining a universal feature, which could be used in any case for any task in any paradigm, in detail, we have explained those terms in the paper, but this is the system model that you use. It's a dual autoencoder, a hierarchical autoencoder. First, we extract the spatial features, but the first autoencoder. Then we send those features for a multi-layer transformer between the encoder and the decoder of the transformer in the latent space, we get those extracted latent features. We check the quality of those extracted features by reconstruction error, by MSE, mean square error. We define the threshold above 99%. If the reconstruction error was satisfied the threshold, for example, in this image, you can see the error is almost nothing. So it means the extracted features after the encoder of transformer is something that we can use for downstream tasks like classification, supervised learning, unsupervised learning, and whatever. Otherwise, we have a feedback, and from the beginning, we start to again extract the CNN, extract the special, and then send to transformer. This process will repeat until we get the latent features here to satisfy what threshold we have defined. Then we use those extracted features for whatever we want. We tested this pipeline, our 12 public data set, our 3 different paradigms, motor imagery, 3 data set, ERP, SSVEP, but all of them was offline. Okay. Before getting to the result, I want to discuss a few interesting characteristics of that model. In traditional features, the features are not flexible. I mean, when we use a specific feature, for example, entropy, PSD, for all subjects, for all trials, for our session, we have the same result. But here, for different subjects, we can select how many features, how many those extracted features in the latent space we can use. And we can understand how many features actually valid for the result we get from any. In that example, the latent space has 128 features. For example, here it is an example of motor imagery data set, a public data set. For example, for a specific subject here, when we reduce the latent features from 128 to 14 features, only using 14 features by threshold 60, I mean, now we can only use 14 features and the result almost is the same. It means we have several features repetitive in the extracted latent feature. We don't have such a flexibility with traditional features. So when we can see which feature is not useful, then the training of the model, the latency will improve. This is a very nice flexibility that universal features can give us. Another important point is that this is a real example for quality of a raw EEG and the extracted latent features. This is what we see, but after pre-processing, this is not dirty EEG. After cleaning, we have such a situation, but this is the same data set of our latent features. We tested these 2 pipelines, our more than 15 different data sets in different paradigms and surpassed all state of the art. But maybe you say, okay, still this is offline. What about real time? And what result? How can you use this pipeline in real time? I joined this group in Spain, Valladolid University, in June, 4 months ago. They have a great BCI lab under supervision of Professor Roberto Hornero, senior leading in the field. And the most important point about this university, biomedical engineering, group, their BCI group, is that they have their own BCI platform. This is very important. Medusa is a very well-known platform for brain-computer interface. And I work directly with the main developer of Medusa, Dr. Eduardo Santamaria. Okay, with their collaboration, we defined a real protocol for semantic EEG decoding online. The protocol is here. It is in Spanish because all subjects, we use 10 PhD students subjects, all Spanish. We designed 4 classes. Turn on the lights. I have pain. I'm in pain. I need some water. I need to go to the toilet. In both image and text modalities. Okay. After getting the insight of several professors in different groups of the university, in the same university, we, as the first step, we designed this protocol. And the detail of the protocol is this. So we have both image and text modalities for each class. Each run has 8 trials. First, the subjects see either an image or text for 4 seconds. This is the perception phase. After 4 seconds, there is a cue, and they speak what they see loudly. This is the over phase. What they see, either the image or text, they repeat exactly the same. It takes 4 seconds. After that, with another cue, with the same image or text, they imagine in their mind. So it means they speak it silently. So we have a perception and imagination phase. And after 4 seconds, we have a rest time for 3 to 4 seconds. For every trial, we have 15 to 16 seconds, and each run takes something like 2 minutes. We use the same for 10 subjects. And for every subject, we recorded data for 240 trials. Then we used our 2 pipelines, first in offline, then we use the same, the trained model, the pre-trained model for 4 subjects in online. Okay, the result is this. Here is an example of the EEG data. This is for the first phase, for the overt phase, when they speak what they see. This is the audio, the voice of the subject. This is the single channel of the EEG. We use 64 channels for EEG and 2 EMG here exactly for recording the muscles during the speak. So we have the audio in this phase, the EEG with 64 channels, the EMG with 2 channels, 3 sources of signal. Also here, the covert phase, the imagination. So we have signal for each class from 4 sources. We used several different approach, machine learning, deep learning, several different traditional features. And the best result that we got, please. Okay, it worked, okay. Before, I have to add something. We removed the last 1 second from each phases because based on, I recorded the data personally based on my visualization. Most of the subjects in the last second didn't do anything. The recording maximum took 3 seconds. So For cleaning data, we removed the last second and including the first 3 seconds for each phase. The best result that we got with traditional machine learning, we got 38% with support vector matching and 49% with transformer. This is after few weeks training different models. Then we use our pipelines. These results are what we use in online after training the model, our 10 subjects in offline, and then giving to the online prediction. In the online, we don't have this phase. We don't have any queue. We don't have anything over the screen. So subjects don't see anything, but they know we have 4 classes. So without overt phase, they think about one of those classes, and the model has to predict what they think about. So the best result we got with traditional features and machine learning and deep learning is good. But when we use our models, the topo map based that we introduced got almost 80% accuracy for binary classification. Binary, it means either the subject thinks or speaks, overt or covert. This is, maybe you say it is simple, it is a binary test. Still, we couldn't get any result with other model like this. But just we wanted to test our topo map based. Then we went to the more complex semantic prediction. This is state of the art to the best of our knowledge. For class prediction, those 4 classes that we shown, it is almost 70% accuracy, AUC, average AUC, our 4 classes. This is not about the concept. The most similar work, what we have found here in online, is a Japanese research with 4 similar concepts, not tasks. It is about 50 or 52% accuracy. These are online, not offline. In offline, we got 95% accuracy. With 4 subjects, those 4 subjects didn't participate in the offline test. So this is valid because there is no data from those 4 subjects in the training of the model. Okay, this is the EEG part and the feature extraction part. Now we have the decoded intention. How to transfer those data safe and securely. For this purpose, okay, yeah, I go. This is completely about different domain. It's about wireless communication, so I won't go to detail. Just I want to briefly mention a nice and crisp usage of what we did. There is a domain in wireless communication named covert communication. According to the covert communication, if we have a channel, a noisy channel, AVGN channel, noise is not something bad. We can use it for hiding the signal, actually. Okay, when we care about the detectability, we don't want a warden, an eavesdropper doesn't understand even the existence of the communication. Okay, according to the covert communication rule, which introduced first time in 2013, in this case, if we have n channel uses, maximum we can send square root n bits. So it means in the classic sense of communication, the Shannon capacity is 0. And it is nice. The Shannon capacity is 0, but still we have communication. Okay. This is something after 12 years, researchers couldn't increase this rate. With the collaboration with the first author of this paper in transaction and information theory, we used our pipeline for semantic feature extraction. How? I had a chance in my second research visit this summer in Braunschweig University in Germany. In the first day I joined them, I met Professor Bolat Bash, the first author of covert communication 12 years ago. After two days discussion with him, I said, okay, the problem of this approach is that if we have a long message like this, we have to split the message into several micro shares, and therefore the latency is a challenging problem. How do you want to solve that? He had no solution. Then this is the problem. Yeah, we have to split the data into several different micro shares, and therefore the latency, the delay is a challenging aspect, and we have to find a solution for that. I discussed with him, this is the baseline when we have to split the data into several different, several micro shares. If we want to use covert communication, why not using an overt phase and several different covert shares, micro shares? In this case, if we have L micro shares in the baseline, the L prime in the proposed solution is much lower than L, and therefore we have a chance to increase the rate. Okay, it means the solution is like this. If we have the same EEG trial, we can split it exactly like what I did, but in the baseline we have to send every share separately. But now we can send an overt share and several micro shares. Then in a state of all shares sending covertly, we send an overt. But the nice aspect of this idea is that in this case, we share some part of the data in the channel, in the overt part of the channel, in the budget of the whole channel. It is clear, it is the warden, the really can seize it, but he cannot understand, he cannot translate what that part alone means. And this is the point, because thanks to the semantic features that we have, decoding that part alone is not possible if really has no access to the covert microshares. In this case, we can increase, this is the solution, we can send the overt part in any case we have the channel, why not use it? We have this power for the overt phase. So we just waste it. Why not using the channel to send some part of the data overtly? Really can seize it, can see this part, but it is impossible to understand, to translate. Why? Because he has no access to the other parts. From other side, Bob has access to both of them, because there is an agreement between Alice and Bob, which is the authorized receiver, and by contrastive learning can pair the microshares with the overshare and understand the whole meaning. But there is an uncertainty, the confusion for the VLE, which is something like gamma bigger than 0. Then that uncertainty, that confusion helps us to increase the covert rate. And here is the result. We paid something like 2% for detectability. We got 20% accuracy, the increase of the rate. This is something very important in information theory. This paper is, this result is in the final steps of internal review, and maybe next week will be submitted. So, next steps. We have to design better protocols with more classes, stronger data, real-time. The latency of the LS, all those BCI platform like Medusa and our model, maybe it is something like 200, 300 milliseconds. We are working to reduce the latency. The generalization for zero-shot transfer, we used only 4 subjects, but maybe we need more subjects. And the robustness, the accuracy is not something we believe valid in this case, because we are talking about a complex task. I need some water. Accuracy is not really a good metric for evaluation such a complex task. And for the transmission, we are thinking about more complex channels like fading, because we use a simple channel, just AVGN, and finally going to the clinical domain. So, as a conclusion, Professor Messine introduced the challenge that traditional features has. We proposed 2 solutions, both offline and online. We got state-of-the-art results, and we used the same extracted intention and semantic also for the delivery, for this communication. So we actually completed the end-to-end flow, not only rely on EEG decoding, preprocessing, or feature extraction. We also introduced a novel approach with the same semantic features for the communication. And thank you so much.
Thank you. Very interesting topic. There are questions? No question. I have a question, but you think about the use of a graph neural network?
No, actually. 3 years ago, a colleague of Professor Messine from Switzerland sent us a paper, and I worked for a few weeks on that paper. I suggested her a solution about graph neural networks, but unfortunately, she didn't share her data. So the solution is stopped. We have no data to test that, but I worked for a few weeks on graph neural network. After that, no. That was the only time I worked on it.
Thanks. There are other questions? No? Thank you.
You're welcome.
Thank you. Okay, the next and the last speaker is Professor Pietro Arricò. He is an associate professor in bioengineering at the Department of Computer Control and Management Engineering of Sapienza, University of Rome. His research focuses on brain-computer interfaces with particular emphasis on passive BCIs for real-world applications such as cognitive workload monitoring and human-machine interaction. He has authored over 120 peer-reviewed publications, holds patent, and has been recognized among the top 2% scientists worldwide by Stanford University and Elsevier. And in addition to his academic role, he serves as Chief Technology Officer at BrainSign, bridging the gap between neurotechnology research and market application. He will add the talk, Passive Brain-Computer Interfaces from Research to Real-World Application.
Thank you. Thank you so much, Stefania, again for the presentation and also for the invitation. I have a hard task, of course, to not make you sleeping because of the number, no?
Yeah.
So I will try to be fast and not much boring, I hope. Okay. So today I will talk regarding this stuff, let's say passive brain-computer interface. That is a variant, let's say, of a brain-computer interface that has been already introduced, for example, in the previous talk. Brain-computer interface, so we know, is a system that connects the brain in some way with the system, with the computer, with the environment, with the appliance, with everything, no? And depending on the features that we are going to use and the kind of application, we can have different kind of brain-computer interface. For example, one of the first, not the first actually, brain-computer interface that has been developed was for communication and control for amyotrophic lateral sclerosis patients, let's say. So the idea was to use this feature, P300 event-related potential. This is a cognitive potential that we can derive from brain activity when we apply this kind of paradigm. So audible paradigm that could be visual, auditory, with different kind of stimuli. It doesn't matter because it's a cognitive paradigm. If I ask you, please concentrate your attention on the letter B and I ignore everything else, and I start saying A, B, Z, B, O, B. Every time you listen B, you pay attention to this and you see, let's say, so we could derive in some way this feature from the brain. The idea was actually in 1973 to develop this kind of interface that is P300 speller that use this kind of potential. I can ask you, please concentrate your attention on letter N, for example, if you would like to write this kind of a letter, and you cross the rows and columns in which you identify the potential, because on all the other stimuli, other columns and rows, you do not have this kind of potentials, and you can recognize by using this kind of system which is the letter that the patient, the user, was going to pay attention, let's say. This is one very old project, and now today it could be very easier and easier to perform, because the aim of this project that ended in 2012, let's say, was to develop a smart home for all, let's say. This was the name of the project, in which one of the way of communication was brain-computer interface. So, and you could control turning off and on lights, opening doors, and so on. At that time, there was not Alexa or Google Nest. It was too much difficult to make all the framework. By the way, we have also other kind of brain-computer interface with active or reactive control, meaning that in which the user try to modulate by using, for example, external stimuli such as P300 potential, or in autonomy, let's say, try to modulate his brain activity to provide a bit of communication towards the external world. For example, if I ask you, please, close your right, left hand, or move your right or left foot. So different parts, different you can extract different features from the brain because different sides of the brain activate. If I ask you, please try to imagine this movement, more or less, let's say, there will be the activation on the same areas, meaning that you can realize a mouse, for example, or moving a wheelchair or something like this. There are other applications we have seen during the last presentation regarding post-stroke rehabilitation by reinforcing the right patterns in the brain and so on. Actually, there is, so how we decode this kind of information? We already know by EEG, for example, is one of the main techniques to extract this information. Why? We have different advantages. For example, temporal resolution that is very, very higher and higher in comparison with other techniques. For example, fMRI that has a very good spatial resolution, but not too much good temporal resolution. We can have a good spatial resolution, of course, because we can have high density, electrodes, and there will be more than enough to derive the features that we would like to to collect. EEG is not invasive with respect to electrocortical activity, let's say. That is the technique that use Elon Musk. Of course, in that way, we will have a higher noise signal-to-noise ratio, but so we will use this kind of technique, let's say. So it's not invasive brain-computer interface. And also, of course, the cost, let's say, with respect to fMRI, for example, we have a relative different cost. Of course, we have to pay something like a Mercedes, not to buy an amplifier, but of course, it's cheaper and cheaper with respect to other techniques. And this is the EEG, of course. So we derive this kind of signals that seems to be noise, of course, similar to white noise. And from that signal, we can extract this feature we were talking about, so the colleagues were talking about before. We have different, can you start, please, the video there, because previous slide. There is the, yes, thank you. Yeah, we have different drawbacks. For example, we have to ask to the user to wear this funny swimming pool cap with the electrodes, with the gel that enhance the quality of the signal because we enhance the conductivity and so on. So it is not invasive, but a bit invasive at the same time, no? Depending on what we have to do. But of course, by using this kind of techniques, we can have this applications. For example, Virgin Galactic suborbital flights, we as group in Sapienza University have participated to, so you can see the two guys on the left and on the right that wear, let's say, this swimming pool cap. The idea was to, so the rationale, of course, of the study was to try to investigate if there were some impairment, for example, in cognitive resources or ability of the users because of the microgravity, let's say. So we have recorded before, after, and during. So we did this to have the starting point from the missions to Mars, but this is another story. It's just to demonstrate that with EEG you can do this kind of stuff. So again, I will talk regarding passive brain-computer interface. What does it mean? It's not still anymore the user that try to modulate his her brain activity to provide the command towards the external world, but it's the system itself that recognize covert changes in brain activity of the user and correlate these changes to mental and emotional states of the user to modify, as a closed loop, the behavior of the environment itself. Just let's think about, we are, I don't know, driving in a highway during the night, no? Before becoming drowsy, this kind of system could recognize in advance the patterns, no? That relates to drowsiness, and, for example, volume up the radio, no, to wake up the user. This is a very trivial example of passive brain-computer interface. But of course, today this kind of system is very central. Why? Because we are in the era of Industry 5.0. Everything is connected to everything else, no, everything except the user. Let's say. So if we succeed to inform the environment, the framework, the whole framework, to what the user, the operator, is feeling from a point of view of mental states or emotional states, we will close the loop. And of course, regarding human factor assessment, this is a central point. Why? Because the most of the risk, let's say, the most of the accidents in operation and in high risky operational environments like aviation, surgery, automotive, again, are not caused the most of the time from the machine, but they come from the user. So if we are able to measure these human factors, we could try to avoid or to mitigate this probability of accident, of risk. But how we measure the human factor? Normally, we can use subjective measures. I can ask someone, how do you feel? Do you feel overloaded, overstressed? Okay, subjective measure is good if you have a big amount of sample size, so 1,000 people in which the variance of the measure is reduced. And also some states are not easy to be measured, okay? So we will do an example later on to explain this point. Or we can use performance, for example. We measure the performance in the main task. If the performance is good, we could say, okay, the workload is not too much high. So it's not too, so it's not really true, this concept. So coming again, going back, let's say, to the example of automotive, no? If you remember when you took your driving license, let's say, at the beginning, no, the performance hopefully were good, meaning that you respected lights, road rules, you didn't kill anyone, hopefully, no? Performance 100%. After 1 year, you can continue to have performance at 100%, but you use a very low amount of cognitive resources so that you can also text during driving. So you can do, but of course, that's a demonstration in which you can see everybody has a maximum available capacity, cognitive capacity, and then there is the demand of the main task. As soon as we get trained, with that task, we have more space between these demands and available capacity. That is called spare capacity, let's say. If we increase the spare capacity, if there is an emergency, there is more probability to be able to face with this emergency. This is the concept. So how can neuroscience provide benefits? This is the loop, let's say. We have EG, we said, in a passive brain-computer interface concept. We have these biosignal processing. Later on, I will show just briefly some feature extraction techniques and so on. And then we derive some metrics that in literature they are called neurometrics, meaning metrics of these human factors, cognitive emotional states felt by the user while the user is performing a task. And we can provide some objective measures of workload, stress, vigilance, emotion, and so on. Why they are useful with respect, for example, to subjective measures? Because they are objective, so we cannot lie. And so the variability of the measure, the variance of the measure is reduced. We can measure online, with subjective measures. At the end of the task, we ask, how did you feel? Now you can do it, this kind of system online, meaning that you can eventually readapt your system to what the operator, the user, is feeling. This is a typical neurometrics processing chain, let's say, for passive brain-computer interface. You have EEG, in which normally we extract feature-based information, so in frequency or in energy domain, so it's quite the same. So we segment the data in order to calculate this frequency domain. Before there is this second step that is very important, as Luca said before in the other presentation, we have a lot of artifacts in EEG. Ocular artifacts, movement, you talk, everything is higher and higher with respect to the physiological EEG. So you have to, not to remove, if you have, okay, you remove, but you have tried to correct in order to not lose a lot of signals. So maybe I'm blinking too much when I talk. So if you remove all the signals relating to when I blink, you lose the 50% of the signal. And then you have this feature extraction phase in which you have to link these features to what the physiology say. For example, an increasing in workload, the normal induce an increasing in frontal theta activity and decreasing in the parietal alpha activity. You select this feature and then you can use, for example, a classification step, a machine learning step, in order to specify that model to that specific user. With all the drawbacks that have been highlighted before, we have this cross-subject, that doesn't work very well because we do not have time invariance of features and so on. Okay, out of the lab use cases, I will show some applications very, very briefly in some research projects that we have done in the last 10 years, more or less. This is a project involving air traffic controllers, let's say. And what we did, you will see an air traffic controller that is managing all these airplanes on the screen. And below, you can see this model of passive brain-computer interface for assessment of overload. What we did was, okay, if the workload is in a normal state, he continued to do his work. If it goes in overload, this school that is an ACC, that is a school in Toulouse where our traffic controller get trained, realized this adaptive interface that was replying to our index, meaning that if the controller was in overload, there were some adaptation solutions that helped the controller, let's say, to decrease the experienced workload. And then we have demonstrated in this paper, of course, the performance, the overall workload, and so on improved, of course, if this passive brain computer interface was used. Then this is another project which we used this concept in another way, just to show how you can do. You can use to adapt your environment. You can use to assess a new technology, an interface, something like this. What does it mean? For example, you have to test a new smartphone with respect to another smartphone. Performance, again, 100%. You reach all the pages, you do everything as you have to do. But with that technology, you put too much effort. So it's not good because you have to decrease your workload because of what we said before, spare capacity. What we did here was to realize this neo-ergonomic study on a new technology in comparison to a standard one, in which performance were the same, but with the new technology, we have more spare capacity. That was good. This is another project. There is a video, but it's not useful. I will explain the project itself. In this project, we developed this metric, let's say, to measure stress. In fact, with a lot of imagination, the name of the project was stress. What we did, actually, there was this operational activity in which we, the experimental task, let's say, lasted 60 minutes. Every 15 minutes, we changed the number of stressors, let's say, in order to stress the operator. So the stress I was saying before, not all the human factors are can be possible to be assessed subjectively. Why? For example, stress is one of these. If I ask you, are you stressed? You can say yes or not, but what you recognize is the stressor, not the stress that induced that stressor. Just to provide an example, if there is a lion behind me that, I feel that I felt stressed because of the lion. If the lion disappeared, I say I'm not stressed anymore. But in literature has been demonstrated in physiology that the effects of stress are maintained over time for 10, 15 minutes, depending on the kind of stress, if it is emotional or something like this. What we did here was the first 15 minutes without any stressor. Second 15 minutes slot with few stressors, noise in the room, telephone calls, and so on. In the third slots, maximum number of stressors. For example, white coat effect. There was an expert behind that started to judge the work of the operator. During the first slot, we removed everything, and we have asked to the user, to the operators, please rate your stress. You can see the first image in the center, let's say, in which there is the statistics. You will see the 4 slots. This is a subjective measure in which we have asked to the controllers, please rate your stress. In the 4th slot, you will see a decreasing in stress. As I said before, the lion disappeared. You do not feel stressed anymore. By the way, by using these neurophysiological measures on the right, you can see that in the 4th slot, the stress is still high. So what was the truth? Then we measured the performance achieved by the controllers during the task. That is the, let's say, the figure below. And you can see that even in the 4th slot, performance were lower and not similar to the first slot. Why? Because there are some effects of the stress that have to be taken into account. Okay? So this is just to show another limitation, let's say, of subjective measures. So maybe this system could be ready to be used for out-of-the-lab application. Are we there yet? Actually, one of the issues, as I said before, is the AG cap. Nobody in operational environment will wear this bulky cap with gel, especially wires and so on. But because, as I said, we are in the new era in which everything is connected, there are a lot of companies that are going to develop even good, I have to say, so you can find different stuff, let's say, but even good headsets that are more wearable, usable for out-of-lab application without gel and so on. I will present now, just to show you, this is an headset, a wearable headset that we have developed as brain science in the startup company of Sapienza within a fast track to innovation project. The aim was to develop a passive brain computer interface system as a whole. So we have developed it from one side. So because we are engineering and we don't know how to do design, we have involved in the IED, so the Institute, the European Institute of Design. Let's say we have an industrial designer now still inside the company that work on this stuff. We have shared with them all the specifications, and they have provided some designs. At the end, we chosen this. We have chosen this because it is wearable, comfortable. There is no gel. There are like sponge soaked with saline solution, so they're not make dirty, let's say, the hairs of the user, and so on. On the right, you will see that processing steps I said before, with all the algorithms that we have developed and fine-tuned to be used for out-of-the-lab application, in which actually they are embedded in this web app. Let's say everything is running in a cloud. So the system is sold, is on the market, let's say, since few years. And you will see these neurometrics that are evaluated in real time, and the user can use it without any technical skill. Of course, this was the aim, and now, as I said, is on the market. And this was before and after. So a younger me on the left in 2014 with this bulky stuff, also with the metrics that were too much dirty. On the right, it's not me, of course. It's the guy that that we use it to publish, let's say, to do market on the products, and also the metrics that are to clean. So this problem is solved, more or less. So there are other problems, as the colleague said before. For example, we have poor control. Normally, with that bulky stuff, you use a meter before each session, let's say, to position in a specific way the headset, the cap, let's say, on the head. Now you cannot do with the wearable device. You cannot ask to use a meter or something like this. No, the operator has to wear immediately, very fast, in a fast way, the headset. But what you will achieve is maybe different features because you are moving the position of the headset with respect to the previous session. So classification accuracy drastically go down. These are just few results, very briefly, on which we are working on. The phenomenon that we have when we move the headset is called normally covariate shift, linked to machine learning approach, let's say. Meaning that we have loose drop in accuracy because we have the feature space that move in another space, because we are just moving the headset. We have developed, let's say, a method to identify this covariate shift occurrence, because we don't know if the headset was positioned the right way or not, and then to solve the problem, let's say, to translate again the feature space of the covariate shift of the wrong, let's say, headset position to the first position. We are finalizing the second step. Now I will show you the results just of the first step, the identification of this phenomenon. What we did was simulating this covariate shift by taking into account a high density, let's say, EEG recording, in which we took closer electrodes in order to simulate, let's say, this phenomenon by simulating just moving the recording electrodes, or moving the reference, or moving both of them, and so on. You will see, for example, in this image, this is accuracy over time, let's say, of the BCI system. You will see the blue line is the intra-session accuracy. It's very high. And then the other three lines are, if I move just the electrodes, and the reference stay there. If I move the reference, but the electrodes stay in the same position. And the last one, the orange one, that is the lowest accuracy, if I move everything. So I move the recording electrodes, I move also the reference. You can see absolutely the drop that you have in accuracy. What we use was this method. We have tested a lot of methods. Of course, this was the most promising one, using isolation forest, that is a machine learning model that works on one class, and it finds anomaly. It's an anomaly detection method, let's say. It finds anomalies in the data. So we calibrate the method by using original session. In the new session, if the space is the same, the method says, okay, there are not anomalies. Otherwise, it will recognize anomalies in the data. And we say, okay, we have coverage shift. In that regard, we are finalizing this new method based on cross correlations, more or less, in which we solve the problem. So we translate again this new space in the original space. And these are just the correlation accuracy between drop in accuracy and increasing coverage shift identified by this method. Okay, of course, there are many other issues when you use wearable device. For example, low number of channels, you have to correct blinks. So normally you use an EOG electrodes in order to use regression methods, for example, when you have low number of electrodes. In this regard, we will not do, because we do not want to be invasive. In that regard, of course, we developed methods to use directly EEG channels to remove this blink contribution and so on. So you have a lot of problems to solve to go from the lab to out of the lab application. Okay, this is just one of the last studies that I show that is regarding teamwork efficiency assessment on surgeons. This was actually a project founded by Sapienza that we did together with Umberto Primo Hospital, let's say. And actually we involved, so the aim of the project was to assess the efficiency of the teamwork by analyzing, also by using graph theory and other methodologies, the synchrony among the brains. As you can see, these guys are wearing this headset, and this is a good point, meaning that with the wearable headset, we don't only go outside of the lab, but we can do a lot of experiments in lab that otherwise you could not do. Just imagine they are doing a real operation here, a laparoscopic operation. Nobody would accept to wear the swimming pool cap with wires, with gel and so on. No, you can do a lot of stuff. And the idea was to have these surgeons that are identified as a kind of graph, in which each surgeon is the node of this graph, let's say. What we used was, because we have a low number of channels, we didn't use hyperconnectivity or something like that that needs to have a lot of EEG channels. What we did was evaluating the neurometrics regarding some affective and cognitive states that are at the basis of the teamwork, something that we did by exploring psychological models and so on. And then we combine these features by enhancing the common information that is derived by the team by using mutual information theory. So we are collaborating with different groups to rate, let's say, to develop this kind of metrics. So by using mutual information, we fuse, let's say, all these metrics of the whole team in order to derive one metric that derive information regarding this efficiency of teamwork. That, of course, provide good results. For example, this is one of the validation in which you can see on the left so green points are teamwork index evaluated on the real teams that were cooperating. On the right, the red dots were related to random teams. So we took different people and we mixed just to test if the index was working. Of course, because it's an index related to teamwork efficiency, it replies better when the teams are real. And there is the interesting result is this: if we compare our teamwork index on the epsilon on the y-axis, let's say, with respect to this physiological scale that is at the basis of the teamwork efficiency based on behavioral and subjective measures, we can see a good correlation, meaning that as the teamwork increases, let's say, also this scale says that the teamwork is increasing. So the results are interesting. So we just have published a paper on this on Frontiers Networks Physiology. This is just, I guess, the last, it's a promise, I think, the last example. Can you start, please, the video again? Okay, this was just to show you how these neurometrics work. This is a project ended in which we have to develop a mock-up, let's say, for a pilot school here in Rome, Urbe Aero Training Center. Why? Because before doing, so after doing training sessions, they do the briefing. They 500. Okay, you can see, so we replaced the same interface, we have the video so that they can do the briefing, and you can see 3 metrics for each of the pilots. This is a commercial flight, a training for a commercial flight. We have 2 pilots, one that really flight or land or take off or something like this, the other one that provide rules, double check, etc. No? You can see 3 metrics for each of the 2 pilots, okay? Workload, so quantity of, so the number of cognitive resources that you put in doing the task. The second is one, stress, how much are you stressed from that task. The third one is cooperation. In fact, is so this teamwork efficiency metric that is the same among the 2 people because it's 1 metric for the team, no? And you will see green side in which they are truly cooperating. The white side, they are not cooperating. If in the true side, you can see an increasing if they are cooperating more and so on. The pilot on the right, so what we simulating here was an emergency landing. There was fire in cabin. They have to ask for a mayday, and doing an emergency landing, okay? But we have destroyed the instrumentation. There was cloud outside, so it was the worst condition, or to be a bit stressful, no? And this is the final part, let's say, of the simulation in which they are going to land, but they are still within the clouds. They don't know if they are in the right line strip, in the right direction, no, to land safely. So you can start, please, again. Okay. So as you can see at the beginning, the water potential is quite high. No? Also the traffic, the right. 100. Okay, now there is the runaway. They go outside the cloud. They realize and you get the stress, the right, and go down. 50, 40, 30, 20, 10. So this was just an example on how actually these metrics can work and the information that can provide. In this regard, we don't want to replace, of course, the trainer, but we would like to do an additional metrics to judge better the training and improve the training and so on. This is the last slide. This is my daughter, 2 years ago, my first daughter. So she's wearing the headset. I say bye to everybody.
Saluta.
Okay. So this was just a funny image to show you how actually by using this kind of new devices open up some possibilities that were not possible to do with standard cap. Just imagine to us this. So my daughter is particular, but she didn't want to wear the bulky system. So these new technologies, apart going on towards out of the lab application, could be very good to do in lab application in different experiences that were not possible to do before. It was everything. Thank you so much again.
Thank you very much. A very interesting talk. If there are some questions, it is very interesting because it seems that anyway, you know which region controls some.
Yes, exactly. Something like this, because the different lobes in the brain, of course,
and also the metrics. For
also
example, the stress. How do you measure stress?
Exactly, because we did so the way which we develop and validate new metrics is at the beginning started with a bulky system with 64 electrodes, something like this. Doing some controlled experiment in which we have to have all the psychological scales, questionnaires, evaluation from subject matter experts, a lot of stuff. And then we try to identify the brain regions within the experiment together with the physiology that is already published and so on, which are the features that would be more responsive to that phenomenon. Then we try, of course, to validate these features also in out-of-the-lab application. So if you start doing something else, but I put some stressful event, does these features still work or not? And we have to go back and modify again, etc. And of course, we use also machine learning techniques in order to fit very well that features to the specific subjects. And then what we did was that the most of human factors that we are using is, so you can retrieve information from frontal sites and parietal sites. That's why at the end we have developed, for example, this headset with 5 frontal sites and 3 parietal sites. But our idea is that in this headset, we have an amplifier that you can click on. It has like magnetic contacts and click on. Why? Because you cannot do an headset that cover all the brain regions, no? Otherwise, you come back to the bulky device. So the idea is develop a different headset. In fact, we are developing, for example, one integrated in VR, in Quest 3, another one for BCI, and so on. You have different headset and just one amplifier that you can click off and on, because it's also the most expensive stuff, let's say, that you have to buy. This was the idea. And this is the routine, let's say, to develop and validate.
Okay. Thanks.
Just one question.
Yeah.
Like, what's the autonomy of the wearable? Like
the wearable is now 3 hours,
3
4 hours. It depends on the sample rate. It can go up to 500 hertz.
Mm-hmm.
If you record a 500, you have 2 hours of recording. 250, that is more than enough. You have 3 hours, 3-4 hours. 125, 5 hours. So, and then you have to recharge. But of course, because we use sponges instead of gel, so they are soaked with a specific design that did our designer, they get wet for 3 hours, more or less, as soon as you wear.
Okay.
So after 3 hours, you have to refresh a bit these sponges and recharge the amplifier. But yeah.
Okay, and what about the quality of the signal compared to the
quality?
So it's acceptable.
It's exactly the same gel-based. Why? Because gel-based, within the gel, there are ionic families, saline solutions related to the iron, to the that you use for the electrodes. Here we use the same saline solution, but instead of having in the gel, why use gel? Because it has to stay there. In this regard, we replaced it with these holders that contain sponges, but at the end you have still water with the same saline solution. In fact, we have demonstrated, there is a paper that we have demonstrated, that we have published a few years ago, in which we have demonstrated the correlation is like 0.99. It's exactly the same for this reason. And that's why we didn't choose dry electrodes, because still dry electrodes has poorer quality. Why? Because you're replacing these means of conduction, that is the salt, that translate for ionic current to electronic current, that is something that you measure, with pressure, meaning that the quality is poorer, and after 10 minutes you have tattoos and you feel pain and you have to remove and so on. So for now, this is, I get wet-based systems, is not just ours, but also other kind of system, is the best solution that you can have. It's just a matter of design, how to increase comfortability, wearability, duration of the battery. So this is the...
Okay. Thanks all the speakers for their very, very interesting talks. And Of course, thanks to all attenders who join us online, and okay, it is a all, and goodbye.
Thank you.
Thank you.
again.
Thank you.